everyone. Welcome back. To all those who joined us for the course, welcome back. But if you weren't with us for the past couple of days, I'm Dr. Sean Gibbons, a faculty member at the Institute for Systems Biology, or ISB. ISB is a nonprofit biomedical research institute located in Seattle, and we are an affiliate of the Providence Health System. We focus on some of the most pressing issues in human health, including, of course, the microbiome, but COVID-19, brain health, aging, cancer, and many other chronic and infectious diseases. ISB also focuses on improving K-12 STEM education and beyond, in particular by addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn more about ISB by visiting our website, isbscience.org. You can see back here. As a nonprofit organization, we rely on generous support from donors. We would like to keep these kinds of events going for free. So those of you who have the means, we'd really appreciate a little help for maybe next year's course. There's a donate button at the bottom of our website. I'd like to acknowledge that we're on the traditional and unceded lands of the coastal Salish people, including the Duwamish people, past and present. We honor with gratitude the land itself and the Duwamish tribe. And before we begin, I'd like to again express our thanks to our sponsors who helped make this symposium possible: Illumina, Metabolon, and the Washington Research Foundation. You can learn more about our sponsors on our website. Because of their support, we were able to provide this virtual symposium free of charge to over 1,000 registrants from more than 70 different countries. All of you tuning in, we're again grateful for your time today. If you're on social media, please remember to share your experiences throughout the 2022 ISB Virtual Microbiome Series using the hashtag #ISBMicro22. And if you want to tag ISB, so we give you a little retweet or reshare, our handle is @ISBSCI, ISBSci. So today we're excited to present some of the leading researchers from around the world who work on sort of broadening the representation of microbiome research to a global sort of population, expanding beyond Europe and the United States, essentially. So as some brief background and on this symposium and how it connects to the course, over the last couple of days, we learned various skills, data analysis skills, looking in particular at populations, 3 indigenous populations from South America, Africa, and Asia. So today you're going to hear from people who are actually doing this research and doing it well, working equitably and fairly with justice in these different communities around the world, and really putting their efforts towards expanding representation in the science that we do. We have 3 sessions today in our symposium. So just some brief introduction to those sessions and what you're going to expect here. Session 1 is called Diet, Lifestyle, and the Global Gut. It's going to be chaired by Dr. Christian Diener, who will be very familiar to you as the instructor of our course, Day 1 course. And the speakers in that session will be Doctors Mathilde Poirier and Gregorio Irola. The second session is called The Curious Case of Prevotella. And that session will be chaired by Dr. Noah Rappaport, one of our TAs and today's session chair. And the speakers in that session will be Doctors Catherine Luzapon and Dr. Cecilia Jimenez. In the third session, this is going to sort of focus on equity in microbiome research. How do we define health in these other types of populations that aren't North America or European? And this is going to be chaired by Dr. Catherine Stevenson. And our speakers today are Drs. Catherine Amato and Dr. Amber Benezra. Each talk will be 30 minutes long. There'll be 2 talks in a row, back to back, followed by a 30-minute panel discussion that's moderated by the session chair. So each session will be in that structure. If you have questions for the speakers, please submit on the Zoom Q&A. At the bottom of your Zoom, there's a little Q&A button. Click on that, you can enter in your question. You can upvote other people's questions, so we can kind of triage questions and pick the ones that most people are voting on as being the most relevant. But when you're posing your question, please do not forget to add the name of the speaker to whom you want to ask that question at the front of your question, and then write your question. There's also the Slack channel. For those of you who are in the course, you know that well. There is a Symposium channel on the Slack. We want you to ask your questions in Zoom, not in Slack, so please don't submit questions in Slack. But Slack is a place where you can discuss among yourselves about how cool the talks are, or you can interact with the organizers and so on. So for more kind of casual interaction, you can use the Slack channel, but for Q&A, please keep it to the Zoom. Just as a reminder, all presentations will be available for attendees to view online on ISB's YouTube channel after today, and they will live forever on our website for this year's course, which will stay up. If you have any questions for the organizers, again, you can use our Slack channel to interface with us. It's a great place to discuss with fellow attendees as well. And I think that's about all I had to say for intro. So I'm excited to see these talks. These are some of my favorite researchers in the microbiome space. So without further ado, I will hand things over to our session 1 chair, Dr. Christian Diener.
Yeah, welcome everybody, and thank you for joining us in this fairly sunny morning in Seattle. It's my absolute privilege and pleasure to welcome Dr. Poirier and Dr. Irola, which will open our symposium with the first session on diet, lifestyle, and the global gut. And as Sean mentioned, just wanted to remind you to ask any question that you have in the Q&A session. It's that little button on the bottom of your Zoom screen. And you can also upvote questions if you would like to increase your chance of being asked. So without further ado, let me introduce our first speaker. And Dr. Poirier, feel free to start sharing your slides while I introduce you. Dr. Poirier is working at the University Hospital Schleswig-Holstein, where she is an associate professor in the medical faculty of the Kiel University in Germany. And she is well known for having authored several keystone papers in the microbiome field. In 2016, she co-founded the Global Microbiome Conservancy at the Massachusetts Institute of Technology in the USA, an international nonprofit initiative collecting the biodiversity of human gut microbes to advance microbiome science worldwide. An ecologist by training, Professor Poirier's research focuses on understanding how human-associated microbes evolve and impact human health. So please take it away, Dr. Poirier.
Can you hear me?
Yes, we can.
Beautiful. Thank you so much. Great. So first, I have to say that I'm officially still an incoming professor. I was hoping to get my appointment started before this talk, but it's not the case. So let's stay, you know, honest here. And yeah, I'm still in transition from my postdoc at MIT. But thank you so much, Christian, for the introduction. Yeah, it's really a great pleasure to be part of this important session today and to talk to you about some of my ongoing work about the impact of industrialization of human lifestyles on human gut microbiome. So before that, I actually would like to make an opening remark about what we know about the human microbiome. The next generation sequencing technologies developed over the last few decades really contributed to advance our knowledge. And this is exactly the topic of our session today, even if we clearly start to know a lot, particularly about how microbiota affects host physiology and how perturbation in the gut microbiome associate with many non-communicable disease, for example, including inflammatory bowel disease, cancers, cardiovascular disease. The vast majority of our existing knowledge is still derived from studies focusing on the microbiome of industrialized population, mostly Caucasian people living in the US and in Europe. And this is actually very well illustrated here on this figure published earlier this year by Richard Abdil from Ram Blackman groups. where you can see the important imbalance that exists in microbiome science. The number of microbiome samples per country and the cumulative microbiome sample per world regions, with most of the sample coming from Europe and North America, and also the over and under representation of each country worldwide, and the annual proportion of sample per region, with again a very important underrepresentation of the less industrialized low and middle income countries in microbiome science. But actually, we already know that the very well represented industrialized gut microbiomes associate with most of the non-communicable disease existing worldwide. As you can see here, when looking at the spread of non-communicable disease occurring more frequently in industrialized areas of the world. So not only the microbiome science is highly biased, but the drawing of the human gut microbiome that we are making is also based on potentially disturbed microbiomes that associate with specific industrialized lifestyles and non-communicable disease. And to end this very short introduction, I would also like to point out that the United Nations recently predicted that 70% of the human population could live in urban areas by 2050, which means that many human communities could soon change their lifestyles and adopt some degrees of industrialization, which could modify their microbial diversity, and with it generate the rise of microbiome perturbation and potentially non-communicable disease among this population. So basically, if we want to better picture and understand the human gut microbiome and its impact on human health, and if we want to counteract the unethical imbalance that exists currently in microbiome science, and that could lead later on healthcare inequities, basically because the underrepresented groups will be less likely to benefit from scientific advances tailored to the most studied population. Well, if we need to, if we want to counteract for all this bias, we need to promote the diversity of human population included in microbiome science, and we need to do it pretty quickly. So for all these reasons, back in 2016, in the laboratory of Eric Alm at MIT, Mathieu Groussin and myself created the Global Microbiome Conservancy, so GMBC. And so this initiative is a non-profit collaboration between more than 80 scientists from 30 countries around the world with very complementary backgrounds, with microbiologists, anthropologists, clinicians, and we are all working together and biobank and make available for research the biodiversity of human gut microbiomes to advance microbiome science worldwide. Equitably and ethically. So together, we already recruited 1,300 participants across a bit more than 40 human communities worldwide. So the yellow dots show the location of all the population we already worked with, and the blue dots show the country where we're starting new collaboration with local scientists and plan to recruit participants in the next few years. So we recruited individuals among both industrialized and non-industrialized countries, as you saw. And in each country, we also recruited individuals among both urban and rural groups. We recruited only adults who do not have any obvious signs of infections or known disease. So the distribution of demographic parameters are shown on the bottom left and are typical of relatively healthy human population. And here I'm also showing you the genetic diversity of our cohort so far, which we characterize by exome sequencing and genotyping, and which we compare to the genetic diversity of the 1,000 Genomes Project. And as you can see, our cohort represents a wide range of populations that have genetically that have been genetically defined so far. So we gathered stool samples from each participant, and we generated multi-omics sequencing data that allowed us to look at really the impact of lifestyles, and particularly the level of industrialization on human gut microbiome. And so on this plot, where basically individuals are colored by subsistence strategies, so the blue dots being the most rural and isolated non-industrialized individuals, and the red dots the more urban and industrialized individuals. And you can see how really lifestyle strongly associate with specific gut bacterial communities, which are drastically different in the gut of people living rural lifestyles in blue, or people living more urban lifestyles in red. So on this plot, based on metagenomics taxonomic profile. And as you can see now, not only the composition is different, but the diversity of bacterial species living in your gut is also significantly reduced with industrialization. And I also wanted to show you these results because the microbiome is not only the bacteria, but many other things. And so I wanted to show you these results generated in collaboration with Malte Hohlmann at the IKMB at Kiel University in Germany, showing that the same trend is also observed when looking at the virome diversity and composition. The virome also highly associates with host lifestyles. So here you have the same color code. And the diversity is also reduced with the more in the most urban and industrialized populations. And finally, so using the stool samples processed in the lab, we created a pipeline to generate high quality biobanks of pure bacterial isolate and their associated whole genomes. So using anaerobic chambers and by designing a wide range of culturing conditions, we built the GMBC biobank, which is the most diverse biobank of human gut-associated bacterial isolates available for research community. And as you can see on this tree, our biobank regroups hundreds of species, including many unknown and many previously considered unculturable bacterial species. And finally, last month, the Global Microbiome Conservancy started a collaboration with another nonprofit organization called Open Biome, and located in Boston in the US, to build up the initiative. So the GMBC program at Open Biome will now be led by Katia Moniz to continue and expand the collection of samples, the biobanking of bacterial strains, and the distribution of biospecimens to the research community. So I highly encourage you to check our website to know more about our work. And if you are interested in joining our effort, please feel free to contact us. But so now I would like to give you two examples of stories that we can go after with these amazing global data sets and biobanks. So the first one about the impact of industrialization on one specific example of host microbiome interaction. So it's a collaborative work with Mathieu Groussin, so the co-founder of the GMBC, and new associated prof at Kiel University as well, and Tu Nguyen with a postdoc in American lab at MIT. And so for this first story, we looked at secretory IgA measured from stool samples. So very briefly, intestinal IgAs are tightly connected to the microbiomes. Once secreted into the lumen, they promote the exclusion of pathogen and antigens. They also interact with commensal cells to prevent overgrowth of biofilm formation, for example. And they also promote the colonization of beneficial bacteria within the mucus layer. So we were curious to know whether the level of IgA secreted, which are directly influenced by the intestinal microbiome, change across population with different gut microbiomes. And actually, when we looked at the level of IgA in the stool sample that we collected from our worldwide populations, we found that the IgA level actually strongly associate with differences in microbiome composition between people, explaining about 7% of the variance, which is illustrated here in this multivariate plot showing differences in microbiome composition between people with individuals colored by a level of IgA. And so the more pink you are, the more IgA you have in your stool sample. And actually considering that usually individual host factor correlate to the microbiome in population-based studies explain about 0.1 or 1% of the variance, this is actually a very strong association. And actually, very interestingly, we also found that this association between IgA and the microbiome composition actually recapitulates the association with host lifestyles. As you can see here, industrialized individuals secreting much higher level of IgA in their gut as compared to non-industrialized individual with a difference of about 1 log. We also observed this difference at a higher resolution, as shown here on the right. So here we compare the IgA level between urban in blue and rural individuals in yellow within each country. And so even at this resolution, we can see significant differences in IgA levels with urban individuals secreting higher level of IgA in their gut. And actually, if you think about it, this trend is counterintuitive, as non-industrialized individuals are actually more frequently colonized by eukaryotic pathogens and parasites, which we actually confirm with our data. So many of these parasites are actually targeted by IgA, but the difference we are seeing here goes the other way around, and maybe related to differences in antigenic nature of commensal bacteria in the microbiome, maybe. So next, we were curious to know if the bacterial species actually coated by IgA in these samples could be different between individuals of different lifestyles. So to check for this, we used a technique called IgA sequencing, which allows for the identification of bacterial taxa through 16S sequencing of the specific cells, bacterial cells that are coated by IgA. And so here, because we had hundreds of samples to check, we use a relatively high throughput magnetic beads-based assays developed by Noah Palm to sort IgA coated cells. And so we calculated an IgA coating index for each bacteria, representing basically the abundance of the bacterial species in the IgA positive fraction over its abundance in the IgA negative fraction. And so we consider taxa with an index higher than 10 to be quoted, or let's say strongly quoted by IgA. And so with this data, we found that the proportion of IgA-coated bacteria is actually higher in the industrialized cohort compared to the non-industrialized population. And this difference being in the same range of as the one that we observe between healthy or disease individuals in multiple diseases, actually, such as IBD, multiple sclerosis, for example. And we also found that the intensity of the coating profile is strongly stronger in the industrialized microbiome, as suggested on this plot on the right. So in conclusion, there is a higher secretion of IgA among industrialized population, with more bacterial species being targeted by IgA and with a stronger intensity. This really suggests that changes in industrialized microbiome composition, also impact host microbiome interactions and our immune system. So we now need to mechanistically understand what's going on here, what's driving this increase of IgA. For example, it could be the results from a higher level of pro-inflammatory signal, or result from higher exposure to gut bacteria in general due to increased impairment of the gut epithelial barrier, for example. So we are currently trying to answer this question with additional experiments, including in animal model. For the second story, I wanted to talk to you about the impact of industrialization on one very specific gut microbiome function, because whether changes in microbiome composition between human population results in differences in microbiome functions is still largely unknown, despite the fact that this is really what we need to know to understand potential impact on human health. So that's a work that I'm leading in collaboration with Mathieu again, but also Enara Sisiaga, from the University of Copenhagen and former postdoc in Roger Simmons' lab at MIT, and Antonio Valencia, postdoc with Ramnik Xavier and Emily Balske's lab from the Broad Institute and Harvard. So this specific function that I will talk about is the degradation of cholesterol driven by the gut microbiota and its potential effect on chronic disease, such as cardiovascular disease, for example. So very briefly, the cholesterol circulating in your intestines comes from, for a minor part from your diet, but also directly internally from your liver, mostly in the form of bile acids. And then about half of this cholesterol is reabsorbed by the intestinal epithelial cells back to the liver, and about half is excreted in stool. But what is less known is that the cholesterol can also enter a convergent pathway thanks to specific gut bacteria. And this pathway converts cholesterol to another sterol called coprostanol, which is only produced by bacterial metabolism. And contrary to cholesterol, coprostanol is not reabsorbed through the lymphatic and blood system, but is entirely excreted in stool. So in theory, if the conversion of cholesterol to coprostanol in the gut microbiota is efficient, then the quantity of cholesterol being reabsorbed back to the liver will be reduced, and a higher level of coprostanol will be excreted in stool. So again, in theory, engineering the gut microbiota so it can convert cholesterol to coprostanol in the intestine, and so reduce cholesterol level in blood, is of great potential in the context of high cholesterol-related disease. And if this works, one easy way to measure the efficiency of cholesterol degradation will be to measure the ratio of coprostanol over cholesterol in stool, because this ratio will be higher when the conversion is basically more efficient. So one of the first things that we did using the Global Microbiome Conservancy biobank of stool samples was to compare this ratio of coprostanol over cholesterol in stool samples. And to make it simpler for this talk, I categorized all the population that we have in just two categories here, rural non-industrialized and urban industrialized populations. And to our surprise, by measuring this ratio of coprostanol over cholesterol in stool sample, we found that non-industrialized individuals have much higher coprostanol over cholesterol ratio than industrialized individuals, suggesting a more efficient conversion being performed by their microbiota. But because these preliminary results could have been driven by many different factors, we then conducted an ex vivo experiment using the same batch of stool samples. So concretely, we put small amount of stool in a tube containing a bacterial media enriched in cholesterol. And we measure the copersonal over cholesterol ratio in each tube over time. And as you can see here on the right, the result we obtain here, for example, after 8 days, confirmed that the microorganisms present in the stool samples of these non-industrialized individuals are more efficient at turning cholesterol to copersonal. So both these first results suggested that gut microbiomes of non-industrialized individuals are better at converting cholesterol, and that this function may have been lost actually during transition to industrialized lifestyle. So naturally, we then wanted to identify the very species capable to carry out this metabolism. But historically, cultivating copersonal producing bacteria have been very challenging. As of today, only one single bacterial strain named Bacterium copersonoligenes and isolated many years ago from pig sewage is available for research. And only one gene of this conversion pathway encoding for a cholesterol dehydrogenase and named ISB has been characterized using these non-human associated bacterial strains. So using the sequence of this characterized ISB gene, we run a genomic screen using the Global Microbiome Conservancy bacterial isolate collection of whole genomes. And we were very excited to identify for the very first human associated bacteria and bacterial cholesterol converter candidate in our biobank. So this isolate is a Firmicutes, genomically very close to the E. copersonoligenes strains and possess a highly conserved dehydrogenase gene in its genome. Using the metagenomics data that we generated from each stool sample, we analyzed and we confirmed that actually the stool copersonal over cholesterol ratio is positively correlated to the abundance of our candidate cholesterol converter. That's a good news. And we also were able to highlight that our cholesterol converting candidate is less and less abundant in a population that have higher degrees of industrialization and urbanization in their lifestyle. So as you guess, we next tested this specific candidate strain in vitro. So it was about the same setup as the ex vivo experiment, but this time we used individual isolated bacterial strains in presence of our cholesterol-enriched media. And so actually, we were extremely excited to confirm the efficiency of our candidate bacteria to turn cholesterol to copersonal in vitro, with the ratio of cholesterol decreasing over time while the copersonal increased. In parallel, we tested bacterial species from 32 different genera, which don't possess the ISB gene, and none of them actually show any sign of cholesterol conversion. But then to look for additional candidate cholesterol converters in the lab, we used our ex vivo approach using complete microbiome communities as described earlier. And so stool samples were incubated with cholesterol, and we integrated longitudinal metagenomics data with metabolomic data to detect taxa associated with copersonal formation. And you can see here on the right the list of these detected taxa that associate with the copersonal formation. And so first, we were able to recover our previously characterized copersonal former, so EcoPro in purple, so that's good news. But we also found a group of species shown here in orange that are all part of the Ocylispiraceae family. So this is pretty exciting because it's one of the unique group of bacteria that has been found to be associated with obesity in meta-analysis of microbiome sequencing data. And so we then looked at those species specifically. And so we grew this Ocylispiraceae species alone with our cholesterol-enriched media to check whether they were efficient at producing copersonal. And actually, when comparing the absolute concentration value to those obtained previously with our EcoPro candidate strain, we measure only, as you can see, only a very little amount of copersonal. But however, when we grow both taxa together, so EcoPro and Ocyli together in co-culture within the same tube with the cholesterol-enriched media, we now see a huge depletion of total sterols in the media. So it looks like the production of copersonal is not that good, but the total sterol, including cholesterol, level drop drastically, about 2 log in concentration, and is actually almost completely removed. Okay, so yeah, we have ongoing experiments, obviously, including the characterization of this co-culturing effect to optimize the cholesterol conversion, RNA-seq generation to characterize the other genes involved in the conversion pathway, and also very promising in vivo experiment that actually seem to highlight the reduction of both fecal but also serum cholesterol in mice fed with our bacterial strains. So yeah, I'm going to end with that, but I think altogether these results strongly suggest that our lifestyle not only influence the composition of our gut microbiome with specific bacterial species missing from the gut microbiome of industrialized population, but also has impact on the presence or not of some function that can be particularly relevant for human health, like this one related to the degradation of cholesterol. Okay, so I would like to thank Mathieu Groussin, who co-lead with me all the work that I presented today, as well as Eric Alm, all the ANLab members, all the GMBC consortium members and participants. And I would like to thank our collaborators that help with the cholesterol copersonal GC and LCMS measurements from Rami Xavier Emily Balskus lab, particularly Doug Kenny and Antonio Valencia, and people from Roger Simmons' lab, and particularly NRS again, Fatima Hussein. Yeah, and I thank you all for your attention, and I see already a lot of questions, so happy to answer in a few minutes.
Thanks so much, Dr. Poirier. Yeah, and just a quick reminder that you continue to ask questions. Just make sure to indicate the name of the speaker as well, so we will know who the question is directed at. And for that, you can use the Q&A section at the bottom of your screen, and those will be answered in the panel discussion after the next talk. So after this amazing kickoff, let's keep the ball rolling with our next speaker, Dr. Irola. Feel free to share the slides while I introduce you. So Dr. Irola is a group leader of microbiology genomic laboratory at the Institut Pasteur de Montevideo in Uruguay, and adjunct professor at the Universidad Mayor Santiago de Chile, as well as an affiliate at the Wellcome Sanger Institute. He is passionate about translating scientific advances into solutions that help sustainable human development while reducing global inequalities. To this end, Dr. Irola has developed and coordinated several large-scale national and international projects, such as the Latin Biota project and deploying the first real-time COVID-19 genomic surveillance program in Uruguay, just to name a few. So please take it away, Dr. Irola, and we'll be happy to hear what you have to speak to us.
Okay, thank you. Are you seeing my screen now?
Yes, looks great, and we can hear you as well.
Right. Thank you for the invitation and for the introduction as well. So I will be talking about our work that has been focused in the last 2 years in unlocking and understanding the composition and structure of the human gut microbiome in underrepresented populations, and in particular in Latin America. And this is motivated by the fact that we all know that the genetic and taxonomic diversity of the gut microbiome is still unsaturated. That means that every new population or every new group of people or cohort, when we analyze them, is composed by newly discovered species that we didn't know about the existence before. So this is particularly relevant, and sorry, because these first slides are gonna be a bit overlap with the previous talk from Dr. Poyet, but I think it's worth mentioning that today the availability of metagenomic data, in particular from the human gut, is extremely biased towards the northern hemisphere, and in particular to high-income countries. And this leaves many entire regions of the world, like those that are highlighted there: Latin America, Africa, Eastern Europe, Middle East, Southeast Asia, as black holes of information regarding the human microbiome. And of course, this impacts our understanding of the role of the human gut microbiome in health and disease. With the aim of contributing to mitigate this bias, we we started our project. And after that, Dr. Poyet showed some figures that were published in this very nice paper that indeed highlighted this preliminary results we had from the screening of public datasets. We put our emphasis on Latin America, of course, because it's from it's the continent in which I got born, and I am from Uruguay, and I developed my science here. But Latin America itself is a very interesting continent, because it's a kind of crystal of different cultures, different levels of urbanization, different socioeconomic levels of different countries. We have had different waves of immigrations across the year. We have a Western lifestyle, but there are still, for example, indigenous communities in many countries, and it's very vast in different aspects. But also from the health point of view, we have a high prevalence of many inflammatory and non-transmissible diseases like cancer, and particularly obesity. Latin America has one of the highest obesity rates in the world, and this is going to make sense at the end when you see some of our results. That I want to mention that when I was seeing the results from Dr. Poyet previously, I was very excited, because the results you are going to see in my talk, I promise, were not premeditated, but they match a lot with what was shown previously. So for studying the Latin American microbiome, I created what I call the Latin Biota Consortium, that today is composed by more than 10 countries. Here I am highlighting the institutions and the countries that originally conformed the consortium, and these are 8 countries: Mexico, Colombia, Ecuador, Brazil, Bolivia, Chile, Argentina, and Uruguay. And from all these countries, we managed to gather information and samples in the beginning. So the phase 1 of our work was from 2019 to 2021, in which we created a multi-kingdom catalog of the variation of human gut microbiome in populations from these countries. We managed to collect over 600 samples from 8 different countries, and most of these samples were from healthy adults, and we also got samples from urban lifestyle and rural lifestyle, and also in very specific cases we got samples from some native isolated communities from some countries. So we perform shotgun metagenomics over these samples, and we analyze them using a genome-centered approach, building assemblies and mags from viruses and from bacteria mainly. So focusing on the bacteria, with this data set, we improved the quality of hundreds of previously described bacteria from the human microbiome, but also we discover an interesting number of new species, new bacterial species that didn't match to any previously known bacterial species inhabiting the human microbiome. So to understand the differences and the prevalence of these newly described bacteria, we compare our Latin American dataset with a global dataset consisting in over 30,000 gut metagenomes. And focusing on this new bacteria, there are more than 300 bacterial species discovered in Latin biota. In panel A, you can see the distribution of these bacteria across the phylogeny of known bacteria from the human gut. But was more interesting for us was to understand the prevalence of these newly assembled and discovered bacteria from Latin America in other continents. And we found that the highest prevalence of these 300 novel bacteria was in Africa. So we uncovered this connection between the African and Latin American microbiome, and we hypothesized that this could be related with the lifestyle and with the less industrialized lifestyle that is a general pattern in these two continents. And when we analyze the taxonomic assignment of this newly discovered bacteria, we found that the first and top taxa represented among them was the order Ocelospirales. And this order Ocelospirales also, when we calculated the prevalence of the Ocelospirales in the different regions, we found then that they follow a vanish-like pattern, in which these species in particular are more prevalent in less industrialized population and vanish from more industrialized populations, mainly from high-income countries. So we decided to explore a bit more the population structure of the Ocelospirales order and the Ocelospiraceae family in particular. Indeed, these plots are from the Ocelospiraceae family. So we perform a population structure analysis and compare the functions that were enriched in those lineages that were more prevalent in Latin America with respect to the rest of the world. And one thing that was interesting for us in this moment was the different mechanisms that were mainly composed by AMR genes. Most genes that were more abundant in Latin American Oscillospiraceae, sorry, were AMR genes. So we perform a more in-depth characterization of the AMR genes across the whole microbiome. And we found that Latin America is the region with the highest prevalence of AMR genes in the gut microbiota. And most of these genes are from the beta-lactamic and tetracycline classes. That also makes sense with the antibiotics that are mainly used for human treatment. Many species, including many of them from the Oscillospirales order and Oscillospiraceae family, were significantly associated with the rural-urban lifestyle within Latin America, and this can be shown in this plot. So we started to uncover some relationship between the lifestyle, the composition of the human microbiome at the global and continental level, and also other important features like antimicrobial resistance and potential reservoirs for antimicrobial resistance. But also, as I mentioned, we focused on the viral species, and here I am basically talking about phages, and comparing against the current databases of phage species that are present in the gut microbiome. We uncover over 9,000 new viral species from Latin biota from this data set. We also show this strong relationship or correlation between the viral diversity and the bacterial diversity. That, of course, makes sense because there is some ecological equilibrium between the phages and their hosts that are bacteria. But interestingly, among those bacterial hosts that were mostly represented among the newly discovered phages, we detected the Oscillospiraceae. So that means that many new phage species that are being detected in Latin America are infecting this Oscillospiraceae family that seems to be very important for any reason in the normal gut microbiome from Latin Americans. Indeed, it is one of the taxa that is mostly associated to the Latin American region if you perform an association analysis based on the abundance of these bacteria in our guts. This is more on the viruses. There are different taxa and different species of viruses, but the main point here is the Oscillospiraceae as one of the main hosts for these new viruses. Also, we did a bit more on this and compare the prediction power of the viral and versus the bacterial component of the microbiota in terms of geographical origin. So we also uncovered that the viral composition of the microbiome has a more powerful signal to predict the geographic origin of a sample at the regional, but also at the country level. So this is something that should be explored more in depth, but is important also from the biomarker discovery point of view. And finally, I wanted to share with you some results about the overall coding and functional potential that is coded by genes present in the microbiota of Latin Americans. And here, basically, we uncover over 200,000 new protein clusters that are new proteins at the 90% level of identity with any previously known protein, and 30,000 of these 200,000 are completely of unknown function. We couldn't annotate anything, any clue about the function of this protein. So there is a vast genetic reservoir in there to be explored and to define which functions, molecular or biological functions, these enzymes, these proteins could be playing. So in conclusion, the phase 1 of Latin Biota contributed to the discovery of over 300 new bacteria and 9,000 new viral species. Novel species are mostly related to the Oscillospiralis and Oscillospiraceae taxa. We also, here there are some highlights about some results I didn't mention due to time constraints, but we developed a new bioinformatic method for detection of viral species with higher accuracy. We uncover for first time the presence of some phage lineages in Latin America. Also, we contributed to enhance the quality of reference gut bacterial genomes, some reference genomes from gut bacteria in public datasets. We also showed the possibility of predicting the origin of a sample from gut metagenomic data, and the virome here is the more informative component of the microbiota. And also the results I just mentioned about the proteins, the new proteins in there to be understood, and also the highest prevalence of AMR genes is something that caught our attention, and is something that we will keep exploring in the future. What's coming next? Well, we have just got some funds from the Pasteur Network to expand the concept we developed in Latin Biota to other underrepresented regions. And in the next few years, we will be working in particular with countries in Northern Africa and in Eastern Europe to gather information about the microbiome in these populations, with particular interest in the role of these populations as reservoirs of antimicrobial resistant genes. Also, we are expanding Latin Biota. We have a second phase in which we want to expand the geographic, ethical, and lifestyle origins of the samples. We are welcoming new collaborators, and we are very excited also to start collaborating or discussing with the people that is today presenting and participating in this incredible symposium. We also plan to explore other body sites. We are very interested in the oral microbiome, also collecting saliva and fecal material from the same individuals, and also integrate our metagenomic data with other sequencing technologies that can improve the resolution at the strain level, or the recovery of mobile genetic elements using this. This is going to be using long reads and other approaches like proximity ligation sequencing. And of course, now we have a baseline of variability of the microbiota at the continental level in healthy population, and we now we want to start analyzing what is happening with different disease cohorts that are very important for the countries that are conforming the Latin Biota Consortium, and those diseases that are important at the continental level. And that's it. This is my group at the Pasteur Institute, and this at the right are some of the people from Sanger that has been helping a lot during these years in every step of Latin Biota. And these are my contacts and social networks. And so thank you very much for your time. And I very happy to answer any questions you can have or any comments.
Thanks so much, Dr. Irola. And a massive thanks to both of our speakers also, Dr. Poirier and Dr. Irola, for those amazing talks. We have quite some time now for our panel sessions, so I think we can get through quite a good amount of questions. So please feel free to keep them coming in the Q&A session, but also upvoting questions that you would kind of like to ask me first. Yeah, and with that, maybe I'll start asking a few questions. I will be mostly going by upvotes and chronological order, but Dr. Poirier and Dr. Irola, if you see like a question that I might be overlooking that you really want to answer, also go ahead and pick those out. And maybe start with a question for Dr. Poirier. And that question was by Noura, who said there was a very interesting work, and they were wondering if the IgA enrichment could be caused mainly by the differences in the diet, or also whether those could be confounded maybe by some environmental factors that are specific to industrialized communities, for instance, air pollution.
Sorry, I missed the beginning of the question, but we're talking about IgA, right? And what's triggering the differences?
Whether the enrichment IgE could, like, what are the main driving factors of the IgA differences? Might it be diet, or could it also be pollution, for instance, industrialized?
Sure, I think it can be very multifactorial, for sure. But our guess, but we obviously, that's just, ideas, hypothesis, and we have ongoing experiments and still a lot of work to do, but our idea is that the antigenic profiles actually contribute a lot to these differences in IgA levels between individuals and between lifestyles. But it's definitely multifactorial, and yeah, why not? Yeah, diet, sure. Pollution, yeah, sure.
Okay, sounds good. Then we also have a question for Dr. Irola. That one is by Sean, and he's asking, is the human use of antimicrobials correlated with the frequency of AMR genes across continents, or do you think this could also be driven by agricultural or industrial usage of antimicrobials?
Well, this is a very, very important question, and it's quite difficult to answer with the actual data, because the data from prescription and or antibiotic use in many countries, particularly in low and middle income countries, is scarce, and some countries gather that information, some others not. So at the country level, it should be difficult to perform in-depth comparisons. But we know, for example, that in Latin America in particular, that is basically food production continent, and where livestock industries are very prevalent. During many decades, antibiotic use was totally uncontrolled in these industries, and antibiotics were used during many decades as growing factors. So they were administered in the diet of animals, and of course this contributed to the selection and evolution of different AMR mechanisms that probably are today present in our guts. But it's an excellent question, and I don't have the answer yet, but probably we will be focusing on trying to answer this with actual data in the next few years.
Yeah, thanks so much. Well, there was one question by Ken. It didn't indicate the speaker, but I think it might actually be relevant to both of you. So both of you can feel free to answer that. And actually, the question was echoed a few times by other participants, and it was basically, are there any studies of differences of infant microbiomes in different underrepresented communities? While there may be differences in the frequency of breastfeeding, there may also be significant differences in the microbiome transferred from the mother.
Please, Gregorio, feel free to jump if you want.
Okay. Indeed, this question, I was just reading it in chat because within our dataset, we have a cohort of in particular, these samples are only from Mexico, but from Mexico, we have a cohort of people from the DF, from the capital. These are urbanized people, and a cohort from an isolated native community called the Mepas, that they live in semi-isolation. And within this community, we have samples from adults and from children, and we have the data from the co-housing of these people. So we know which families live together. And this is something that we didn't analyze. We didn't have the time to analyze Mexico as a whole and perform these comparisons, trying to look if these children in this community that are mainly feed through breastfeeding exclusively, and they are not exposed to other to medicines, for example, or to antibiotics, have differences in their gut. We did some preliminary analysis in comparison with other cohorts of children across the world, and including other rural or native communities. And in particular, we found some interesting patterns with the Bifidobacterium species that are those that start to colonize the gut. And in Mexico, the Bifidobacteria species that start to colonize are different from other industrialized and also non-industrialized communities in different parts of the world. So there are some species that are very prevalent that are not prevalent in other parts of the world, and vice versa. So it's something we have there, but we didn't have time yet to explore more in depth. Yeah.
And we don't have infant in our own cohort, so yeah, we will not. But we confirm that in the most isolated and rural areas, most of the children has breastfed for many years actually, and there is a lot of co-founder effects obviously, but that's definitely have impact on the gut microbiome.
Perfect, thank you. Yeah, then we have another question by Sean. That one is directed at Dr. Poirier. And he's saying, really interesting results. Any idea about the ecological pressures that enrich for coprostanol converters in non-industrialized populations? I might have expected the reverse due to what I'd expect to be an enrichment in cholesterol in the industrialized diet.
Yeah, that's a very good question. First, the enrichment in cholesterol in industrialized diets. Our data, so we have dietary information from all the participants in our studies, and using this data, we don't really confirm a huge difference in cholesterol enrichment in industrialized diets. Actually, a lot of very rural and isolated population consume a lot of meat or eggs, for example, several times per day. So that's actually a pretty cholesterol-rich diet. So that's one thing. About the ecological pressures, so that's only hypothesis because we didn't test for that yet. But one hypothesis that we have is that so the EcoPro strain that we isolated and tested in vitro, we actually realized that it's extremely sensitive to oxygen. So one explanation could be that the gut of the most industrialized population may be more exposed to a higher level of oxygen. If it's more leaky or it's a little perturbed or whatever. So that's the hypothesis that we have, but that's just ideas. We need to test for that.
Perfect. Thank you. We have another question for Dr. Irola by an anonymous attendee, and they asked, do you plan to look at parasites and fungi at some point? Has any progress been done in culturing any of the newly discovered bacterial species from Latin America?
Cool. Indeed, regarding the eukaryotes, we did it. We did a attempt to assembly and characterize eukaryotes from our data, but we didn't found much things except for some blastocystis genomes that we were able to assemble and characterize. And we suspect this is because biases in sample processing and mainly DNA purification, that some eukaryotes are more difficult to, DNA from some eukaryotes are more difficult to extract. And also the relationship between bacterial and viral DNA and eukaryotic DNA in terms of the absolute quantity may bias the thing towards the bacteria and viruses. So we couldn't achieve much results on this, but it's something that is very interesting because there are very, very few analyses of the microbial eukaryotes of the human gut, and probably they are playing a very important role that we don't know. And regarding the culturing, yes, I didn't present this, but we are also generating a catalog of and a biobank of bacteria cultured from at least some of the samples. We couldn't have access to culture from all these countries, but indeed we are doing this now, and we already have a collection of over probably 2,000 strains from Uruguayan population, and we are doing more. But now we are indeed very interested in focusing on culturing Oscillospiraceae species and have a look at some phenotypic characteristics of these bacteria that are very prevalent in Latin America.
Perfect, thanks. Yeah, I'll give you a short chance in case you saw a question that you really want to answer, but no need. I didn't want to put you on the spotlight here. I'm happy to select questions. It's just I just want to give you the explicit chance in case I'm skipping something. But yeah, while you look through that, also there was one question to both speakers. There was, what do you think is the relationship between the virome and lifestyle geographic location? Is it about viruses that infect and modulates the microbiota, or does it also include host viruses changing along with the microbiota and diet?
Yeah, really not a specialist here. I don't know if I want to make up something, but
no, well, what we found about the virome was surprising for us, because we expected to identify novel viruses, but the number of new potential phage species that we uncovered was like tremendous, and they vary according to geographic location. But in particular, we don't know why. There are some populations that we analyze that are very, very high in novel viruses. For example, population from Bolivia, we don't know why, is extremely rich in terms of new phage genomes among our data sets. And well, we don't have also metadata to hypothesize why this can be happening, but it's a result that is there. There are some populations that are much richer in viruses, and the virome seems to be much more volatile across populations and across individuals, and this is why probably viruses have the potential of being very powerful biomarkers for certain regions or certain populations. about the mechanisms of how viruses can modulate. There are different mechanisms. Also, the viruses can modulate our immune systems directly. So they can act over the bacteria, controlling certain bacterial taxa, but also directly have an effect on some immune pathways. So it's very vast. Yeah.
Perfect. Maybe I'll misuse my position as a chair a little bit here and maybe butt in with a question of my own as well. So hearing both of your talks, I was also wondering about the environmental factors as well, such as many participants. So when obviously the microenvironment in the gut itself is probably responsible for a lot of differences that we observe, but then there's also like, environmental factors that affect the host itself, which again can propagate to the gut microbiome maybe. Do you think it would be necessary to maybe also start sampling the environment itself, for instance, sampling particularly foods or dietary components from different regions, or taking like air samples or any other samples from the environment where those communities live to kind of get maybe at it? Or do you think that, questionnaires usually capture the majority of that variability?
Well, Matilde, do you want to?
Sure. I mean, I don't.
Okay.
Yeah.
No, sorry. No, go ahead if you.
Thank you. So yes, to answer the end of the question. So yeah, no, I think even with tons of questionnaires, we're missing a lot of information. So yeah, I will really love to see more environmental samples. So I'm aware of quite some groups that started to check the microbiome of dietary incomes, and particularly fermented food, for example, source of water. That's also very important. And yeah, I think that's an important next step. I think I'm missing the point of the question, but
no,
was that it? Yeah, good.
Yeah.
Well, indeed, we have a project in which since some years ago we are doing metagenomics on the urban waters in our capital city, basically sewage, and we have a paper that is probably out in a couple of weeks. It's the preprint available if anyone wants to read it, in which we analyze across 2 years how the microbiome from the urban environment is influenced by human microbiota, and how this impacts on the dissemination of AMR. And we found very interesting patterns in the dissemination of AMR genes to carbapenem antibiotics, correlated with the development of different outbreaks in hospitals, in which the patients are delivering part of their microbiome through the pipes, and this of course reach the environment and have an epidemiological impact in how our closer environment is composed, and how it could, in the future, can impact in our health. So this is something that we are doing, but also the water sources or other sources of different microbiotas could be very interesting to analyze together with the human microbiome.
Yep. Perfect. Thanks.
And actually, when you look at the species composition in the gut of the most isolated and rural population and individuals, you can already spot a lot of species that are clearly environmental bugs. Actually, so we need to work on that, and we're probably missing a lot of these species that are maybe not exactly, well, not exactly human gut microbiomes, but just, environmental bugs just passing through because, less sanitation or, source of water and diet, for example.
Yeah, perfect. Thanks so much for answering that. And we have a question from Sunda, which I will take the liberty to reformulate a little bit. So what are the possibilities of harnessing microbiome metabolism within human hosts and alleviating, mitigating in general? Maybe I will ask for more general like disease or like improve health, especially in underrepresented communities.
Is that for me? Because I'm not sure I get the question, sorry.
Oh, sorry. What are the possibilities of harnessing the microbiome metabolism, or maybe the work you have done in general to maybe improve health as well in those communities?
I see.
Mm-hmm.
I think diet is a good start. Improving the source of complex fiber, for example, will be a good start. We know how it's important for many bacterial species that are missing in the gut of industrialized population, and species that potentially have high impact on human health. So yeah, I think that could be a good start.
Yep, Dr. Irola, feel free if you want to answer that as well, or
sorry, because I was trying to answer another question.
Oh, sorry. Yeah,
in
just
the chat, but
yeah,
no problem.
I do the same. It's not easy to follow.
yeah, sorry.
That's exciting.
Yeah, the question would be also for Dr. Irola. Do you think that some of the work you have done, for instance, on the Latin Biota project could be like leveraged to maybe, create more specialized interventions to the microbiome, specific for Latin America, for instance, so that like intervention that would work better for that population than, for instance, trying to just extrapolate from, known things that we know work for like Northern American or European populations.
Yeah, indeed, this is our longer term goal, to provide a resource and a new knowledge that can be used to improve regional or local interventions that can be adapted to what is normal for our populations. And for that reason is also that in this second phase of the project, we are starting to focus on the analysis of different diseases that are prevalent here, because, for example, we can have microbial biomarkers for certain diseases that are not the same for Latin American people that for African, European, or US populations. So yes, the answer is yes.
Yep, perfect.
I spotted a question that I didn't answer yet, if that's helpful.
Yeah, definitely. Go ahead.
From Segman, what could explain the improvement of cholesterol removal by changing from single culture to co-culture? The hypothesis, again, that's just an hypothesis that we have. So when we used just the EcoPro, so the first cholesterol converter strains, we actually very quickly, after a few days, usually 7-8 days, we reach a plateau. Even if you had more of the strain, we reach a plateau and we cannot turn more cholesterol to coprostanol. However, when we add the Oxidase spiraceae in the co-culture, then both sterols and all the sterols in the media decrease. So the hypothesis is that actually the first PC turned cholesterol to coprostanol, and the second one used the coprostanol generated in the media and reduced this to something else, not a sterol or not something that we can detect, but that helps to, continue the process and the metabolism and to reduce the overall sterol content of the media. That's the idea. We gonna test for that easily by replacing the cholesterol by coprostanol in the in vitro experiment. So hopefully we will have an answer for this question pretty soon.
Perfect. So we have a question by Sean again to both speakers, and he's asking, how have you thought about partnering ethically and equitably with underrepresented population in your research?
That's very, very important aspect, I think. And in particular, we didn't yet, but we are open to do it and also keen on doing that. Of course, if there is some idea or how to reach this, I would be very, very grateful to hear, because it's something that we didn't do because we prioritize the core activities like sampling, sequencing, analyzing data, and indeed we are now working very hard to get the first paper published from this whole data set, but in the longer term this is very important, and it can help the project itself a lot.
Yeah. So on our side, so I think there is a lot of levels where you can actually be careful. So I'm just giving an answer of explaining what we do. I don't say that's what we should all do. We're trying to do science the right way. We're trying to do the best we can, but we are also very open to any suggestion, and we're open to learning more about the best practice. But I think the first very important thing is to avoid what's called parachute science. So I think having collaborators in each country where you actually work is extremely important. You have to have local collaborators, local scientists in each country, because they know the country, they know the population, and they should also be part of advancing the microbiome science from their respective countries. So that's the first step. Then what we do when working with underrepresented population, so indigenous population in some case, saying that the consent form is an important step. We strongly believe that having consent form, written consent form in the local language is extremely important to give as much information as possible to the participants, so they know exactly what's going on, why we are here, trying to collect some samples from them, etc. And that's also on the same topic, what we are trying to do, and we just started in a few countries, but we really hope to be able to do that in all the countries we work with. We also think it's important to get back to the population after you actually do the work, after you actually generate the data and you the results, and you publish the paper. You have to go back to the population. You have to go back explaining what you found and why participating to this study was so important. And I think it's just trying to be respectful to the participant and everybody. There is many aspects that could play a role here. I'm probably missing a lot, but that's already an overview.
No, that's perfect. Thanks for the replies. There were two questions for Dr. Poirier, and they're both related, basically, which mouse model was used and whether those were germ-free mice or were treated with antibiotics to level the microbiome.
So I'm certainly not saying that one model is better than the other, but so far we used regular mice after antibiotics treatments. That's what we did, but there is pros and cons in both models. You should not just listen to my answer, just see with who you are collaborating, see if using a model or another can help answering your question, but
Yeah, perfect. Yeah. And I also had another question for Dr. Irola. And in the beginning of your talk, you also alluded to that Latin America is maybe one of the continents, one of the most complex migration histories as well. So how do you think that is kind of, I mean, you already showed some migration patterns that were obviously, but do you think that's like, has a particular influence on the microbiome maybe compared to other continents or populations?
Well, I think it has indeed. We performed some global scale analysis with the whole dataset, in which we found strong signals and strong correlation between the distance in terms of microbial diversity between individuals and their geographic distance. So that all supports and adds more on the concept that different regions in the world and different populations have their own patterns of variation, and so the migration patterns can affect that. There is some very interesting paper, I don't remember, it is published in Cell, I think, I don't remember now the authors, about how the different generations of immigrants from Southeast Asia to the US, their microbiota is altered generations after regeneration from the date they immigrated into the country. So we didn't measure for our data set because we don't have much information about the ethnical or genetic background of the participants and the donors. This is a weakness of our first data set, but for sure it is important, and there is something there. And Latin America is very complex. We have had during centuries a very important influence from slavery from Africa. Also in the Pacific coast, countries like Peru have or had an immigration, a very strong immigration from Japan and from Asia. So there is very, very complex. And today, what we found probably is a picture of all this mixture across many years. And also these immigration patterns have influenced a lot today's diet in these countries, because for example in Peru or in Uruguay, our regular diets are absolutely different.
Yeah, thanks so much. So I think we have now taken almost 40 minutes in the panel session. So I think I'll give our speakers their freedom back and thank them so much for taking all the time with us, and it was really very illuminating, amazing talk. So a huge virtual applause again to our two speakers today. And yeah, I think that was a really great start for the symposium. We'll be having a short break now, and then we're starting with our session 2. Thanks so much to the speakers again. It was great having you.
Thanks again for the invitation.
Yeah, thank you for the invitation.
Hello, everyone. I am very pleased to open our next session of the symposium, which will focus on the curious case of Prevotella. In this session, we will have two speakers, Dr. Catherine Luzapon from the University of Colorado and Dr. Cecilia Jimenez from the National Autonomous University of Mexico. Time for Q&A will be right after the two talks as in the previous session. So please make sure to deposit your questions in Zoom using the Q&A button at the bottom of the screen. And please do not deposit questions in Slack. Please note that with the speakers, please note which of the speakers the question is intended for. You can also upvote other questions so we know which ones are in high priority. Each talk will be 30 minutes, and then after both talks, we'll have about 30 minutes for questions for both speakers. So without further ado, I'd like to welcome our speakers to this session. It's my pleasure to introduce our first speaker, Dr. Catherine Luzapon. Dr. Luzapon is an associate professor in the Department of Biomedical Informatics at the University of Colorado. She has been heavily involved in the development of popular computational tools from microbial community analysis, such as UniFroc, that we talked about in day one of the course. Dr. Luzapon's lab focuses on investigating gut microbiome composition and function, as well as its interaction with the systemic immune phenotypes and metabolic comorbidities. She will talk to us today about the immune modulation by Prevotella-rich microbiomes in men who have sex with men and potential implications for transmission of HIV. Dr. Luzapon, please take it away.
Thank you so much for the invitation. Can you hear me okay and see my slides all right?
Yes.
Okay, great. So it's a pleasure to talk to you all today about my work. So work in my lab focuses heavily on HIV-infected populations and the role that the microbiome plays in many aspects of HIV disease pathogenesis. So HIV's main target cell is CD4 positive T cells, and the most large concentration of these types of cells in your body are in the gut. The gut is also an important site of HIV transmission because anal intercourse among men who have sex with men is a main source of mode of transmission. And so the degree to which gut microbes influence the activation state of these CD4 T cells is of interest for transmission because activated CD4 T cells expressing HIV co-receptors are more likely infected by HIV. Once transmission happens, it's been shown that there's a breakdown in intestinal barrier function and alterations in the microbiome as microbial products and parts translocate from gut to blood. They activate CD4 T cells, further enhancing infection. With ART, or antiretroviral treatment, a lot of this is ameliorated, but there are remaining defects in barrier function and systemic immune activation that have been linked with metabolic and other comorbidities in this population. So I started researching this field over a decade ago, and our first paper that we published was in 2013. And in this paper, we used 16S ribosomal RNA targeted sequencing to characterize the fecal microbiome of 4 different cohorts: people with recent HIV infection who had not received any treatment, people with chronic or over a year had been infected with HIV but still had not received any treatment, and then people with suppressed virus on antiretroviral therapy, and then a group of HIV negative controls. And in this paper, we saw pretty striking differences in the microbiome. And so in this picture, each of these is a taxa bar chart. So each color represents a different bacterial family. And one thing that was, I think, pretty visually obvious is that the 3 HIV cohorts on the left have a lot of this pink family. That's the Prevotellaceae. And so Prevotella copri was actually the dominant species in this bar. And they had less of the yellow than the controls. And the yellow is the Bacteroides family. And this is diverse species within the Bacteroides genus. So when I saw this result, it was not what I expected to see. I've looked at a lot of other microbiomes and inflammatory diseases like inflammatory bowel disease. And, I was expecting more a low diversity microbiome, outgrowth of pathogens that immune dysfunction might bring. And so when I showed this result, my collaborators, Tom Campbell and Brent on the project asked, well, is Prevotella bad? We see so much more Prevotella with HIV. Could this be a problem? And, my initial answer was, I'm surprised by this result because I thought Prevotella was quote unquote good. And there was actually kind of at the time a lot of mixed messaging regarding the potential role of Prevotella in health in the literature. So around the same time we published this paper, there was actually another paper that came out where they showed an expansion of intestinal Prevotella copri in people with new onset rheumatoid arthritis. And in this paper, they showed some evidence that Prevotella copri could be pro-inflammatory. So for instance, in a mouse model, they colonized the mice, they gavaged antibiotic treated mice with either media or Prevotella copri, and they found that Prevotella copri could colonize and expand, and that when they treated these mice with DSS, which is a detergent that affects the gut and can cause inflammation, it's a model of inflammatory bowel disease, they found that the Prevotella gavaged mice lost more weight, and they had a poor endoscopy score. So there was some evidence that Prevotella could quote unquote be bad. We in our hands were trying to take bacteria that we saw increase and decrease with HIV and do immune assays. So we culture we order the most closely related bacteria from a culture collection, we culture them, and we expose human immune cells to them and measure inflammatory markers. And so here I'm showing the ratio of TNF alpha, a pro-inflammatory cytokine, to IL-10, an anti-inflammatory cytokine. And in our assays, so here's Prevotella copri. The color is whether the blood was from an HIV positive or negative individual. That had no effect. But we found that Prevotella copri didn't really, in these assays, act more pro-inflammatory than Bacteroides species that we assayed. But another microbe I'll talk about later caught our eye in this assay, and that was Eubacterium biforme, which has since been renamed to Holdemanella biformis. So despite some evidence that Prevotella could be bad or inflammatory, I was really slow to kind of demonize Prevotella because I had also been involved in work as a postdoc in Jeff Gordon's lab, where we had been looking at the gut microbiome across people in different cultures. And so we looked at individuals from really young to old age in the USA and compared them to individuals from an agrarian cultures, both in rural Malawi and in Venezuelan American Indians. And this is a PCOA plot of unifrac values clustering. Each point represents an individual, and the color represents where they're from. And you can see on PC2 here, we had very clear separation from people from the US from these two agrarian cultures. PC1, I'm not showing here, but that was age. So we have infants here and we have adults here. And this change across cultures was associated with differences in Prevotella and Bacteroides. So this is that same plot, but now I've gradient colored them by their relative abundance of Prevotella. And so you can see individuals in agrarian cultures have a lot more Prevotella and less Bacteroides than individuals in the US. Some individuals in the US pull out towards the agrarian cultures, showing more Prevotella. And there were other papers coming out at the time that showed that this was actually associated with diet in the US. So this is looking at fecal samples from 98 healthy US adults, and they compared the microbiome, whether people were Prevotella rich and Bacteroides poor, or the opposite, and they call these enterotype 1 and enterotype 2. They related this to surveys of what these individuals typically ate, and they showed that individuals with the Prevotella rich, Bacteroides poor enterotype were eating less compounds that would be found in red meat, like amino acids and fats, and more compounds that would be found in a more agrarian type diet. So there's really good evidence that diet might be driving differences in Prevotella and Bacteroides. independent of any kind of pathology that the rheumatoid arthritis papers were suggesting. Since then, there's also been papers that have come out that suggest that in the context of a high fiber diet, that Prevotella is good for your metabolic health. So this is a paper out of the Backhead lab where they fed individuals either barley kernel bread that was high in fiber or white bread that was low in fiber. And they challenged these individuals with glucose and looked at the spike in glucose in their blood. And they found that among certain individuals, those who ate the barley kernel bread had a protection compared to those who had white bread in this spike. And they also showed that these responders who responded well to a fiber diet had a concomitant bloom of Prevotella. So suggesting that Prevotella in the context of a high fiber diet is good for your glucose tolerance. At the same time, though, there were papers coming out saying, well, Prevotella could be bad for heart disease. So again, looking at Prevotella and Bacteroides enterotypes and showing that people in more Prevotella were making more of a harmful metabolite for atherosclerosis called TMAO from components of a high-fat diet. So when trying to make sense of this, I kind of was thinking, it's not that Prevotella is good or bad. It depends on your diet. So like if you're eating a high fiber diet, Prevotella is beneficial. But if if you have a Prevotella-rich microbiome and you're eating a high-fat diet, it's could be detrimental. And there was actually similar things on the other end of the spectrum. Prevotella and Bacteroides sort of trade off. And there were papers in mice showing that administration of certain Bacteroides uniformis, in the context of a high-fat diet, ameliorated metabolic and immune dysfunction that high-fat diet produced. But these beneficial effects of Bacteroides, again, only occurred in mice fed a high-fat diet, not in mice fed the standard chow. So context-specific interactions of Bacteroides and Prevotella with health, with diet. So what I found interesting was in this other paper that also found Prevotella-rich Bacteroides poor microbiomes in HIV-infected individuals, they related this to diet. And they found that in their HIV-negative controls, there was a very nice correlation between the relative abundance of Bacteroides and the number of servings of red meat per week, suggesting that a healthy person is suggesting for beneficial bacteria in a diet-dependent manner. And this relationship was completely obliterated in their HIV-infected subjects. So that association between diet and the microbiome that helps you process that diet was disturbed. And so based on all of these papers at the time, I kind of came up with a hypothesis that, there's not one healthy microbiota. You want Prevotella if you're eating fiber, you want Bacteroides if you're eating a Western diet. And I thought maybe the reason this gets messed up in HIV is that CD4 T cells have been shown to interact and select for some Bacteroides, and these are targeted by HIV. And so you default to a Prevotella-rich microbiome. So we looked further into this hypothesis, and we recently published a paper on this. So I'm not going to show all the results of it, except to say that, unfortunately, we are not actually finding very strong support of this hypothesis. We did find one amplicon sequence variant, and we had a few that mapped to Prevotella, but one of them predicted metabolic disease in this population, but it was beneficial. So it's more consistent with the paper out of the Backhead lab saying Prevotella could be beneficial for your metabolic health, and not supportive of our in this population, it won't be. So anyway, we did find a lot of other microbes associated with barrier function, which was associated with poor metabolic health. So there were other microbially mediated mechanisms supported in our work. So we could have maybe guessed that our hypothesis would fail, because very early in the study, we realized our hypothesis was flawed. And it was flawed because we made an error that I think a lot of microbiome papers do. And that was, we did not select an appropriate negative control population in our study. So, so we have, since that initial paper in 2013, a few years later, we published another paper that really provides better information on how the microbiome differs with HIV. Rather than looking at just a couple dozen individuals, now we're looking at 217 individuals. And in this table, I'm showing you the degree to which the individuals in our HIV negative, HIV positive, ART naive, and HIV positive ART treated cohorts distributed again across 3 different groups. So we have women, and then among men, we have men who have sex with men and men who have sex with women. So as I mentioned, anal intercourse among men who have sex with men is a major mode of transmission of HIV, and it is very much in the populations in Colorado we're looking at. So if you look here, among our HIV positive cohort, the vast majority of them are men who have sex with men, even more so in our untreated cohort. And so in our prior study, all of our controls were just selected matching for things like age and gender, but not sexual behavior. But in our work moving forward, we began to recruit from a cohort of individuals of men who have sex with men who were at high risk of acquiring HIV. So all these individuals were actually recruited for a vaccine trial. They were having sex with many other partners, unprotected, so high risk activities. And so when we now had a control population with some MSM, men who have sex with women and women, it painted a different picture. So here's another PCOA plot. And here the points are colored by female in pink, men who have sex with women in orange, and men who have sex with men in green. And then the shape is whether they have HIV or not. So circles are HIV negative, and HIV positive, treated and untreated, respectively, are the squares and the triangles. And you can see we have clustering by sexual orientation, gender, rather than HIV infection status. And so, and when we looked, so now here's a plot showing, this is in this, it's a taxa bar chart, but it's they're ordered in the same order as PC1 here. You can see this very striking pattern. The pink is the Bacteroides, the yellow is the Prevotella. As you move from the side where you have the non-MSM to the MSM, you get a loss of Bacteroides and a gain in Prevotella. So this opened up all these questions, right? Like we thought this was HIV driven, and we had all these hypotheses. It was driven because defects in CD4 T cells were disturbing interactions with Bacteroides. So this is not true. So we had to then step back and ask, what is even the difference in the microbiome in HIV when we're controlling properly? And then also, why do the high-risk MSM have this different microbiome? And when we were recruiting this cohort, we actually, had some information on sexual behaviors. We've started asking more questions on that than we used to. But what we tried the best with the data we had. So for instance, this is a figure from that paper where it's the same plot, but these are indicating whether men and women are engaging in receptive anal intercourse, which you would think would be one reason they might have a different microbiome, and it didn't explain it. So we had women who engaged in RAI who still clustered with other women, and we had men who never engaged in the receptive part of anal intercourse, and but were having sex with other men, and they were over here. So at this point, I cannot tell you why the microbiome is different in this population. I can just tell you it is. And so really then, because our biggest microbiome differences were in these high-risk MSM, we just had new questions about, now we had 2 questions about both the HIV-associated microbiome and its effects on immunity, and also the high-risk MSM microbiome and its effects on immunity, because this is a very important population because they are at high risk of getting HIV. And so one of the first things we wanted to do is really just better understand, are there immune differences in our high-risk MSM populations? And we found that there were, and there were some in actually some very important immune populations for HIV disease pathogenesis and HIV acquisition and control. And so in one paper, like we looked at the blood immune markers, and we were specifically looking at ones that people in the HIV world really care about. So for instance, CD8 T cells expressing these activation markers are important markers of morbidity, disease progression, comorbidity in people with HIV. And it has been shown in many papers, it's elevated in HIV positive people, but it was actually also already elevated in our high risk men who have sex with men compared to our non-MSM. And then this is another interesting immune type in HIV, because CD4 T cells are the target of HIV, and CD103 are the percent of those that are, that's a mucosal homing marker. So these are moving, they're moving either to or from the gut, the lungs, something like that. And MSM had more of these than non-MSM. And HIV positive didn't, which actually makes sense because HIV kills these cells. But the fact that they're higher, these target cells of HIV and MSM, is interesting. We also were interested, because transmission is really important in this population, on whether the microbiome could influence the expression of the HIV co-receptor CCR5 on CD4 T cells. This is required for HIV to infect T cells. And again, if it's activated, these are activation markers, then these cells are going to be even more likely to be infected. And we found, using biopsy samples and looking at rectal biopsy samples, that our high-risk MSM actually had a higher proportion of their T cells were expressing these markers that would make them readily infectable by HIV. And then we also, in a more untargeted search, where we actually used technology called CyTOF to screen many immune populations, we found this very interesting population called mate cells was depleted in MSM, whether they have HIV or whether they don't have HIV, compared to our non-MSM controls. Mates are interesting because they're enriched in tissues. They're important in mucosal barrier integrity, maintenance, and repair. And they're known already to interact highly with the microbiome. In fact, they respond specifically to bacterially derived riboflavin synthesis intermediates. And previous studies have reported a loss of mates with HIV and made the same mistake we made. So this has everything to do with HIV, like influencing the immune system. But having this important control population shows it doesn't actually have anything to do with HIV. But this is important in this population because mate cells actually can respond to and suppress HIV. So again, a loss of this in high-risk populations is potentially going to promote the spread of HIV. So the next thing we want to do in these studies, this is all, we found differences in the immune system, we found differences in the microbiome. Are these related to each other? And so to do this, I collaborate with an immunologist, Brent Palmer, to do a lot of lab work. And a lot of the work I'll show you today was done by two different postdocs in his lab, Sam Lee and Preston Neff. And so there's two different types of assays we'll do. One is we take either cultured bacteria or whole bacterial communities from feces, and we use them to stimulate immune cells. I already showed on an earlier slide a result with stimulating peripheral blood mononuclear cells. We'll also get gut cells from leftover surgical surgery tissues. And then we also use notobiotic mice. So rather than look at individual bacteria, like I showed you on that earlier slide with Prevotella and the Bacteroides having similar pro-inflammatory signals, here we are actually taking like the whole feces. And so we're taking a fecal sample, we're using histodense column to get out the whole intact bacterial cells. We check how good our prep is with flow cytometry, and then we also sequence it, and we can show that our isolated bacteria have similar compositions to the whole stool, and we're able to reproduce, bacterial preps that are enriched in Prevotella versus Bacteroides to use in these assays. And so, like, we take the fecal microbiota, expose them to PBMC, and look at flow cytometry of these cell populations. One limitation of this are these microbes are dead. It's not like working in a mouse model, but it's sort of telling you, like, the sum of their parts, how those components of their cells might be influencing the immune system. And so one of the things that we did, remember we had, and we published this in Gut Microbes a couple of years ago, one of the things that we were interested in were these CCR5, because it's an HIV co-receptor. And what we found in these assays was that, so here we have non-MSM, and then we have men who have sex with men who are HIV negative, and then men who have sex with men who are HIV positive, either ART treated or not. And all three of these MSM populations, regardless of HIV infection status, had elevated stimulation of CCR5 on their CD4 T cells, suggesting that what we had observed just looking at biopsy, that these populations were higher, and that the microbiome as a whole was actually a potential driver of this. It supports that it is a driver of it. We also were able to do infection assays. So very similar, you take the fecal microbiota, you expose immune cells to them. Now we're actually doing immune cells from the gut. A surgeon, Martin McCarter, he does gut resection surgeries, usually for tumor removal. He gives us the healthy tissue. We get out the immune cells. We stimulate them with the fecal microbiota, but then we actually also try to infect them with HIV. And what we found here is that the cells, the gut immune cells, T cells that were pre-stimulated with the fecal microbiota of MSM, whether they were HIV negative or positive, were more readily infected by HIV compared to the men who have sex with women controls here, or here's media. So that told us that the microbiome as a whole was having, really important drivers potentially for transmission. And so next, we used the same data that I showed you, where we correlated, where we showed elevated CCR5 positive cells and MSM, and we looked to see which microbes were correlating with this. And we didn't find Prevotella, which was our most dominant microbial difference. Instead, we found Holdemanella biformis as our top positively correlated microbe. And if you remember from the slide, that was the one that we had already tested in vitro and had a really pro-inflammatory cytokine profile. And actually, the only negatively correlated one was a Bacteroides, an ASV mapping to Bacteroides. So it could be the lack of Bacteroides that's also contributing. And I'm not going to go into too many details here, but we recently published another paper where we, took out a couple of strains of, actually cultured some strains of Holdemanella out of patient samples. We compared their ability to induce HIV co-receptors to Bacteroides uniformis, and one strain was higher. They were all trending. And then actually that one strain also had a particularly pro-inflammatory profile, cytokine profile. So we compared their genomes, and they had differences in these capsular materials that could be responsible for these differences in immune modulation. So we're continuing to research, how and why certain microbes could be driving these important phenotypes we see. We also used notobiotic mouse models. So again, we took these three different patient populations, and we looked at this, and we didn't get perfect colonization, but we did get an increase in immune activation with the MSM regardless of HIV status. And we put Holdemanella in the mice, that same species, and we could see that actually would increase it, and it correlated. So we were still getting evidence of the same microbe. So what about Prevotella? Prevotella didn't colonize the mice. And so here's the profile. And you can see Prevotella In gray, it was in the donors and not the recipients. This was surprising because prior studies have shown Prevotella does. We took Prevotella coppery, it did monocolonize, and interestingly, the Prevotella from our HIV negative individuals did colonize, suggesting some sort of strain level variation. And indeed, when we actually look at the variation in MSM, there's a few different strains. These are different ASVs of Prevotella. Most go up, one goes down. And so I think that Prevotella is showing some strain level variation in this population, which would be very consistent with what we've seen in comparing Prevotella from people with westernized lifestyles generally to non-westernized. There's great differences there. It's actually a very complex complex of different strains with not a strong core genome. And there's more strains in the developing world. It's kind of a paucity of strains in the US. And this has been associated with differences in their genomes in terms of carbohydrate breakdown. We are in the process of getting shotgun metagenomic data so we can see what this looks like with our MSM populations. So in summary, populations of people living with HIV who are MSM and high-risk MSM tend to have Prevotella-rich Bacteroides poor microbiomes. We hypothesize this might influence their metabolic health because of a diet microbiome mismatch, but we didn't find evidence of that. We did find evidence that this is more of an MSM phenotype, and this Prevotella-rich Bacteroides population microbiome is correlating with undesirable immune phenotypes. But immune in vitro assays and mouse models are really pointing to some other microbes that correlate with Prevotella, not Prevotella itself, and driving it. And so we've really honestly been looking more at other things, although we are going to look at that strain level variation in Prevotella now, because I feel like that could be actually obscuring the detection of any role if we're just binning all the Prevotella when they shouldn't be binned. So with that, I will stop and thank the people in my lab who did the work. A lot of the analyses were done by Abigail and Janet and Nicole, and also all of my HIV collaborators, our study participants, and the NIH. Thank you.
Thank you so much, Dr. Luzapon. Dr. Jimenez has a problem and she had to drop out from the Zoom. Therefore, Sean Gibbons will be speaking instead of Dr. Jimenez. So let me briefly introduce Sean. Sean came from the University of Chicago and completed his postdoc training in Eric Elm's lab at the MIT. His postdoctoral work focused on developing techniques to quantify individual-specific evolutionary dynamics within the human gut microbiome. He then came to us at the Institute for Systems Biology, and his lab develops computational and experimental tools for investigating host-associated microbial communities to explore interactions between ecology, evolution, and the ecosystem function. He is now working on developing personalized interventions for improving human health and well-being. So Sean, take it away.
Hello, everyone. A bummer that we lost Dr. Jimenez. She just dropped off our Zoom. I'll filibuster for a second here, just in case she pops back on as I pull my slides up. But rather than missing out on our second talk, I guess I'll give you all a little story here on the microbiome and its impact on how we respond to drugs. Let's see. No news. She's still not on. Okay. Well, sorry everybody if you're going to get me. Sadly, I had a backup talk ready to go. Are my slides looking the way they should look? That should be swapped. Okay. So this isn't perfectly in line with the theme of this session, although there will be some mention of Prevotella, and I guess we got a really great overview of the sort of curious case of Prevotella from Dr. Luzapon. So, okay, recent work from our group in the past few months, which was published in the journal Med, has delved into this question of, we know the microbiome can metabolize drugs and can interact with pharmaceutical drugs, but how does that really impact the human side? How does that affect the response of us to those drugs? So we'll get into that. And I don't think we need much introduction on the microbiome, since we've gotten a fair amount of that. I like to say it's sort of changed the way we think about ourselves as entities, right? We're not entirely human. We're never alone, right? We're more than 99% microbe when it comes to the genetic composition of our bodies, right? More, but it's 100 times more genes pent up in our microbiome than there are in our own genomes. And it's a complex system, right? In complex systems, the whole is greater than the sum of its individual parts. And sort of classic 20th century reductionist forms of doing biology aren't as relevant to these types of ecological or system scale phenomena. There's a lot of emergent phenomena that can come out of these systems, and you have to study them at multiple scales and integrate across those scales in order to get meaningful insights out. So again, we know the microbiome is important. It affects all the systems of the body. You can kind of think about it as a metabolic or endocrine organ. It probably affects about half of the circulating metabolites in our bloodstream. But the question we're going to ask here today is whether or not it affects how we respond to drugs. And the drug in particular that we'll focus on are statins. Statins are cholesterol-lowering medications taken by lots of people. I think it's the most prescribed drug on Earth, definitely in the developed world. More than 20% of all Americans over 40 years old are taking these drugs. And they're life-saving, right? They reduce the occurrence of stroke and of heart attacks, and generally they've been a huge boon to medicine over the past several decades. Now, while they do save lives, and you should always listen to your cardiologist and take your statins if they tell you to take your statins, there is an interesting wrinkle to how statins affect our bodies. One of the most common side effects that I won't talk about as much is that they can cause muscle pain, pain in your muscles. And so a lot of people suffer from this, and they have to work with their clinicians to find a tolerable dose of the drug that allows them to take the drug without pain. But the other big side effect that we'll focus on more today is that it sort of messes with your metabolism a bit. It messes with how you process lipids, and this can affect your chances of developing type 2 diabetes. Those who are on statins are more likely over time to transition into type 2 diabetes. It sort of makes you more insulin resistant, essentially. And oftentimes people with diabetes themselves are targeted for statin use because they themselves are more likely to experience heart attacks. So it's usually thought that heart attacks are scarier than diabetes. And so that trumps the fear of progressing towards type 2 diabetes. So usually people are just given statins without much thought to the sort of metabolic side effects of those statins. So this brings us to our study. We were interested in the heterogeneous effects of statins across a population. So this is work that was led by Tomasz Wolmanski in our collaborators lab, the Hood-Price lab, along with my co-corresponding authors, Noah Rappaport and Andrew Magis. Some of the microbiome data processing was done by Christian Diener, who you all know well at this point. And like I mentioned before, Nathan Price and Lee Hood are the collaborative lab that helped out on this project. So if you look at prior work that delves into the efficacy of statins, you see in this, I think it's called a waterfall plot, a huge range of effects to the same exact dose of a statin over the same time period. In about 8,000 people here, you see that some folks have a really, really strong response. Their LDL cholesterol is taken way down, when they take a statin. But in some people, you actually see an increase in LDL cholesterol with statin use, which is not what you want to see. And indeed, the amount by which the drug brings down your LDL cholesterol, that is correlated with, or it predicts your risk for events downstream, for having a heart attack. So you can see that people who had the biggest reduction in LDL have the lowest risk, essentially, for cardiovascular events. There's been some prior work in the microbiome field in statins and the microbiome. This is a paper from 2020 in the journal Nature, where they were focusing more on the effects of the drug on the microbiome itself, right? To taking statins reshape the composition of the microbiome. And this is a European group that often works in what we call enterotypes, which is a sort of clustering method to cluster together similar types of microbiomes. And in the developed world, although this is definitely not the case in the developing world or other parts of the world, but in the developed world, we tend to see 4 clusters. There are 2 clusters dominated by the genus Bacteroides, which you saw in Dr. Luzapon's talk. This is the genus that is dominant when Prevotella is not dominant. There's a cluster that's Prevotella dominant, where Bacteroides is actually very low in abundance. And then there's a cluster where it's enriched more in Firmicutes, or in particular Rominococcaceae, so depleted in Bacteroides. So these 4 groups are distributed across the population. And what they found is that, this BAC2 group is associated with worse metabolic health. So people with higher BMI tend to be enriched for this particular enterotype. And the big take home of this particular paper was that statins appear to protect people from transitioning into this BAC2 enterotype. It was a cross-sectional study, so they didn't actually watch people transition, but they found that there was a slight depletion in this BAC2 enterotype in obese people who were taking statins. So there was an idea that there was maybe some protective effect here. We were actually more interested in the inverse question of whether or not microbes, the composition of your microbiota affects your response to statins, not how much statins affect your microbiome. In fact, we didn't really find much of an effect of statins on the microbiome itself in our study. The reason we embarked upon this question is prior work has shown that if you expose commensal microbes that are in the human gut to a wide range of drugs that humans take, different pharmaceuticals, you find that many of our commensal microbiota are able to metabolize known pharmaceuticals. And so this is just an example. On the x-axis are many different strains of commensal microbes that were cultured in vitro, along with a drug, in this case, lovastatin, which is a statin that I think is the original form of statin, which was derived from a fungus, I think. And you can see that by and large, organisms in the Bacteroides phylum tend to be pretty good at degrading lovastatin. And you see patchily other taxa doing it as well, but the take-home message is microbes can deplete the concentration of a drug if it's present in the gut lumen. And so what are the mechanisms that could be going on here? It's kind of hard to say. There's a lot of ways things could be acting, like the statins could be reformulating or reshaping the composition of the microbiome, which has collateral downstream effects. That's sort of what this other Nature paper was trying to point towards. Microbes can be depleting the amount of the drug that's available to the human body and pulling it out before it gets absorbed. And the statins, their main effect is to inhibit an enzyme in the liver, HMG-CoA reductase, which is the rate-limiting step in cholesterol synthesis. And so maybe microbes can interfere with that inhibition by depleting the drug, or maybe even turn it up by turning the drug into a more active form, into like a prodrug. And another form of modifying the body's cholesterol, like Dr. Poirier mentioned, is that microbes can convert cholesterol into caprostanol, which maybe increases the removal of cholesterol from the body, or they can affect bile acid synthesis. And so that could also potentially be contributing to the balance of cholesterol in the body. So we are interested in kind of seeing the overall reflection of what's going on in the gut, with the ecology of the gut, the metabolism of the gut, how that's reflected in the body, and in this case, how it's mediating our response to drug. So, okay, here's the data set that we analyze. Is this Aravale cohort, where we have a lot of paired information about host physiology, molecular phenotype of the human side, along with the gut microbiome. In this case, we sort of sub-selected a set of people where we knew their statin usage information. So we had about 1,600 non-users and about 250 statin users. And among those statin users, we had really high resolution information on the dosing of the statin they were taking for about half of those folks. And so again, statins are inhibiting this particular enzyme called HMG-CoA reductase. It's actually hard to measure the effect of statins without having a time series. Usually the effect of a statin is measured as the change in LDL cholesterol over time since taking the statin. But we only had a snapshot. We only had cross-sectional data for most of our population. So we wanted a marker that was sort of a snapshot marker for statin efficacy. And we landed upon a particular chemical that we could measure in the blood called HMG, which is essentially this upstream metabolite in this pathway. It gets cleaved from the CoA by an esterase, and it just sits around in the bloodstream. And so we thought that, well, maybe people who had more or less inhibition, we'd see essentially the quantitative relationship with the levels of HMG in the blood. And yeah, we kind of did. It was a little noisy, but it seemed to kind of work. Where in people who were not taking statins at all, there was no relationship between LDL cholesterol and plasma HMG levels. But when people were taking statins, there was a negative association. So lower LDL cholesterol, more buildup of this precursor metabolite in the blood. And overall, there's a population shift towards higher HMG if you start taking statins. So now we have a measure of on-target efficacy of the drug. Just to delve a little more deeply into whether we think it's a good measure of this efficacy, we see that there's a quantitative relationship between statin dose and the level of this marker in the blood. So there's a positive correlation, right? The higher of a dose of statin you're taking, the more you see building up in the bloodstream of this HMG. And it's also associated with LDL cholesterol coming down, right? So higher dose is also associated with a steeper reduction in LDL cholesterol. There are also genetic markers of statin efficacy, SNPs essentially in your genome that determine whether or not you're going to respond more or less to a statin treatment. And we see convergent evidence here as well for HMG as a marker, where we see no differences in plasma HMG levels between people with two different genotypes for high responder and not a low responder to statins when they're not taking the drug. But when they're taking the drug, you can see everyone goes upward in their HMG. Everyone has more buildup of this particular analyte. But people who are predicted to be stronger responders from their genetics see an even greater bump upwards. So this is just further evidence that HMG is indeed a decent marker of on-target efficacy. So what if we look at the microbiome? So when we look into the microbiome, we find that people who have more diverse gut microbiomes show less on-target effectiveness of a statin. So their HMG levels bump up not as high if they're really high diversity versus low diversity. So on the x-axis are the tertiles of Shannon diversity. So the most diverse third of the population, the middle and the least diverse on the left-hand side. And for here we have, I think, both, richness, observed ASVs, and Shannon, and we see the same sort of effect. You can ask whether or not this is robust to dose, and indeed it is for the small subset of people where we have dosing information. And if we control for many other confounders, we find that there is a negative association between diversity and the buildup of this HMG. There could be a lot of reasons for this. We don't quite understand it yet. It makes some sense that maybe if you have more diversity, the chances that you have organisms that are good at degrading statins maybe increases. So let's go back to this enterotype picture. So in that prior paper, they had looked at these clusters called enterotypes. And we were able to cluster our population into the same 4 groupings. So that was convergent between these different data sets. Like we talked about in Dr. Luzapon's talk, there's a group of people who are highly enriched in Prevotella. And so that particular enterotype shows this really bimodal association. Either you have a lot or you have a little. In the Bacteroides groups, you see that they both are enriched for this particular genus. But this BAC2 group that's thought to be less than healthy, it shows a characteristic depletion in butyrate-producing Clostridia, like Faecalibacterium presnitzii, for example. So it shows the lowest abundance of this particular organism. Okay, so we did try to recapitulate this effect that was observed in the Nature paper, that maybe statins were somehow protective of the BAC2 phenotype. We actually could not find that result. We saw indeed the opposite, actually, in our population, where people who were statin users actually were enriched relatively to non-users for this BAC2 enterotype. And this was true even if we looked at the same data sets that this group looked at. However, if we partitioned our analysis and looked at people who were very sick, who had essentially advanced heart disease, those individuals did show a depletion in BAC2, but it only really worked if you subselected the population in that particular way. So this is where it gets more interesting, I think. If we then, instead of thinking about our statins affecting the composition of the microbiome, which in our analysis, they were, there was a significant effect, but it was very, very weak. What was much more striking was that the composition of the microbiome seemed to affect the on-target efficacy of the statin. So you can see this BAC2 group, they show the biggest jump in their HMG when taking statins compared to non-statin users. And the BAC1 group also shows the next biggest jump. The Ruminococcaceae group, they show a significant upward tick in their HMG levels when they take the drug. So they're having an effect. It's not as strong. And interestingly, we didn't have a lot of data for the Prevotella group. We don't have a lot of data points here, so it's hard to know whether this is a really reliable result. But it seems that those folks who have high Prevotella microbiomes didn't have as much of an effect when taking a statin. And this is also reflected in how much you bring down LDL cholesterol. The folks with high BAC2, they brought their cholesterol levels down the most. And this was all kind of controlled for a lot of different confounding variables. Now, what's even more interesting, I think, is that the off-target effects in terms of promotion of metabolic disruption, these were also affected by the microbiome composition. So people with a BAC2 enterotype, which tangentially, I should mention, harkening back to the alpha diversity result, BAC2 is also the lowest diversity of these enterotypes. So the association with the diversity seems to be largely driven by this BAC2 enterotype. Those who have this particular enterotype show the biggest perturbation to their metabolism. So their HOMO-IR is elevated, their HbA1c is elevated compared to everyone else. So they seem to be most disrupted in their metabolism when taking the statin. You see a little bit of that in the BAC1 group, excuse me. But the Ruminococcaceae group appears to be completely protected from this. We could recapitulate all of this analysis in an entirely independent cohort from Europe called the Metacartus cohort. Excuse me, I'm recovering from a bit of a cough. And we see the same exact results. So this Bacteroides association is true in this other cohort. The enterotypes on-target and off-target effects were true as well, which is super exciting. So, this is, I think I'll wrap up with that. This is sort of the first, I think, detailed observation of the sort of host side efficacy of a drug being modulated by the composition of the microbiota. Oh, thank you so much. Here's my water. But we're trying to understand the mechanism still. We don't quite know what's going on. We have some data on blood metabolomics suggesting that certain bile acids in the blood might be modulating these effects. So we see, say, for example, ursodeoxycholate enriched in the bloodstreams of people who have high BAC2 enterotypes. But ultimately, we need to do more work to understand mechanistically what's going on here. And we'd like to run some sort of trial, either a longitudinal trial or even an interventional trial, to see if we can leverage what we've learned to give people precision drug interventions that are optimized for their metabolism and for their bodies. And that's ultimately the dream, right? To be able to leverage this information for precision medicine. So I'll skip this. Christian is actually currently working on ex vivo studies to try to get a mechanistic picture of what's going on. Thanks to the lab. And I will now turn it over to Noah. And I guess I'll keep sitting here because I think that's how we're going to have to do it. Yeah, I actually don't. Where do I? You got it?
I got it. Yeah.
Oh yeah, stop share. Up here.
Thank you, Sean, for this great talk, as always. Also great paper, by the way.
No, you're unbiased.
Okay. Let me start with a question to Dr. Luzapon. So this is a question from Christian. Do you think that some of the negative health impact of Prevotella in low fiber diets might be due to Prevotella compromising the intestinal mucosa? You touched on that a little bit, by using it as a carbon source, maybe in conjugation with other taxa.
I've seen at least one paper kind of suggesting, that Prevotella through mucin degradation, like I've seen people suggest that, but I feel like I've seen a whole lot more papers on Bacteroides, like certain ones, like also, metabolizing host mucins. And so I think that's actually, kind of a complicated question. Like, there's really, in terms of their biology, no reason to think like, oh, Prevotella really degrade the mucins and Bacteroides don't touch it. I would almost think it's the opposite.
Yeah. Thank you. That makes sense. The next question would be to both speakers. Is also from Christian. And how do you think, how conserved do you think are the association between the enterotypes that both of you mentioned across the different communities with different lifestyles and cultures? So do you think it will hold up with communities that are also, that all contain underrepresented populations?
Yeah.
Kathy, you want to start? I feel like you're better.
Yeah. Well, no, I mean, that's a great question. I think there's a lot of reason to believe that they won't be super consistent, because I was kind of running out of time at the end, so I rushed through them. But like there has been work done, even just comparing like the Prevotella species sort of complex and what in agrarian cultures versus the U.S. and even within the U.S. with vegetarian, vegan, and omnivorous diets, and the Prevotella are very different in terms of their genome content and their their carbohydrate utilization gene clusters. And so I really think I really wish I did this earlier. I'm finally like getting shotgun metagenomes on Prevotella rich MSM, but like just that the Prevotella don't colonize mice when they do from non-MSM. Like I do think there's a lot of biological difference there. In the beginning, I was really trying to relate what we saw to all the literature on Prevotella, and the more I learn, I almost feel like we're just dealing with a different animal. It's not, and so I don't really think, I think the reason some of my initial hypotheses turned out to not pan out was because it's just a different, it's different.
Yeah. That's part of the reason we wanted to have this session, is like, it's just constantly intriguing to me how crazy Prevotella is, all these varying associations in different directions. And I'm trying to understand how it works. I think it's just such an exciting place to look right now. From the enterotype side of things, they're often kind of calculated at sort of like a 16S sequencing level, like genus or species level. And if we go back to like the data we had in our course the last couple of days, we looked at the Hadza and a few other populations. you look at the stacked bar plot of the abundances of different genera in those samples, and it's like, it looks like a completely equal distribution of all the there's no one taxa dominating. It's just super, super diverse. If you try to run these clustering algorithms, like the Dirichlet multinomial mixture model thing on these samples, you don't get 4 clusters. You just don't. If you look beyond Europe and the US, the story becomes just infinitely more complicated. I was a little grumpy about enterotypes early on in my career, but I kind of believe it for like America and Europe, that you can kind of squint and lump people into these 4 categories ish. But when you look beyond that, it doesn't look to be the case. And so I might ask the next question, if you don't mind, Noah.
Go ahead.
Because it kind of, I think, tips off from this, like what's going on here and what are the functional sort of relationships here. But Kathy, I might have missed it in your talk. Did you took fecal material from men who have sex with men and incubated that with like immune cells to look at responses. Did you grab high Prevotella folks who were non-MSM and do the same experiment?
Yeah,
you did.
Yeah, we only had a couple Prevotella folks who were non-MSM, so they would have been included. We didn't exclude them, but we didn't really have the power to just take the non-MSM because it was only just a couple. But yeah, like right now we were just looking at that, how many are, because for the shotgun we're currently doing, I was trying to get to some people in our hands who are non-MSM Prevotella rich just to, even, have that. And yeah, it's just a couple people. So.
I see. I'll jump in again while you look.
Okay.
Noah. But another question I had, Kathy, was, the fact that you didn't see a similar effect for women who were having anal intercourse, like that's like mind blowing, right? Like, what is the behavior driving this? And did you take more detailed behavioral information from folks about sexual behaviors?
We did.
You did?
We expanded the questionnaire a little bit, you know what I mean? And like, just to ask about particular things, like there's actually one paper out from Colleen Kelly's lab where she actually, in a very controlled way, had men use a type of lubricant that she thought was going to be particularly destructive to the gut, like a hyperosmolar like lubricant. And like, she did see some changes kind of in that direction with that lubricant, but it just, for me, I just can't explain it all, because unless people are not being honest, there's just men who are like, I'm always only the insertive partner. That's what they're saying, but they have this microbiome. So then actually I noticed someone else was the question, like, can you do strain tracking? Maybe something's driving it, but then, you share there's been stuff a lot done with heterosexual couples showing, you share microbes with the people you are intimate with, and it could be like something's driving it, and then it gets sort of passed, because it really just that someone who engages in anal intercourse but never is receptive, it just throws away anything you'd think of, like, like do you use an enema before? There's all these things that you think maybe that's it, but it's like, but all of them are more associated with the receptive partner, So yeah, so it's been really hard to figure out, and I don't actually think that like our sexual behavior data is that poor, because we found other reasonable things to correlate with sexual behaviors. Like we've seen, like some other pathogens that could be, passed, to those to correlate with people who are, reporting more high risk behaviors. But the Prevotella is honestly just so prevalent that there's just not any single thing that all of them are, everyone with that are doing, it's like most of the cohort has that, that microbiome type. So
I'd love to see a detailed strain tracking study with sexual partners. I know there was, I think Murat Aaron had something with like penis and vagina microbiomes and looking at transmission, but I think like looking at more diverse sexual behaviors and transmission would be interesting. I suspect, I don't want to get too deep into the weeds of sexual behaviors, but as an MSM myself, I suspect there's perhaps potentially more fecal oral transmission opportunities in MSM behaviors. And some work I've seen suggests to me that Prevotella might be actually really, I don't know what the word is, maybe contagious, but if it gets transmitted anaerobically, so to speak, or it doesn't have to travel very far between people, and it gets into a new system, it seems to be pretty good at taking root. There was a study I saw in New Zealand where they had like competitive FMTs where they took donor material from 5 people and they mixed it together, and then they gave it to a bunch of recipients. And one of the 5 donors was high Prevotella. Every one of the recipients high Prevotella after the FMT. Yeah. So I wonder if it's just like one of those things that gets in and just like dominates.
And we have tried to write. We have written unfunded grants to look at all these things, but we were kind of thinking about the same things you're thinking about, that it could be spread, and looking at that. And we also just kind of, we wanted to look at MSM who were not high risk. This isn't all MSM, like that we're looking at. It's a very select group, and and then also kind of, and it'd be hard to recruit, but like younger MSM, newly sexually active, because if that's true, it's just what you would expect, that they would less, that it just gets acquired over time. The longer you're in MSM, you just pick this microbiome up from your community.
Teenagers before they're sexually active, and watch them over time and see if you just see a flip in the MSM. Yeah. That'd be really cool.
That would be interesting, but no one wanted to fund that. No. I tried.
Sean, I have a question here from Javad Sadeghi, and he's asking, is the statin metabolism starts in the intestine, and can statin change saliva microbiome?
Good question, Javad. Statins are often in pill form, and so I doubt they're dissolving already in the mouth, but maybe a little bit. I'm guessing a large amount of the statin drug is absorbed in the small intestine before it even reaches the colon, right? So how much the colonic microbiota are affecting the levels of the drug that's absorbed is kind of up in the air, because maybe almost all of it's absorbed in the small intestine. However, when we look at stool and use metabolomics, we can see statin showing up in poop. And if we run ex vivo incubations, we can actually watch that statin be degraded by the microbiota. So we know that it's there at appreciable levels. We know that the microbes are still munching on it even after it comes out in stool. And so I think it's hard to say how much of the actual microbial metabolism of statin is affecting its efficacy in vivo versus maybe it's some really indirect thing going on. Like maybe it has more to do with bile acid synthesis, and that's being indirectly shifted around by the microbes, and it's not having anything to do at all with actually metabolizing the drug. We're trying to figure it out. Actually, it's kind of a quandary.
Sorry, can I ask a quick that builds on that? But have you looked in your ex vivo, what the statin is being turned into, and then then you can get that spectrum of things and screen whether that's correlating, especially with some of those. You said your back two had more comorbidities, where there are particular metabolites of the statins that you could track and see if that's driving it.
Great question. And we're going to soon Christian just got a grant to do more ex vivos with statins, and I know is involved in that as well. But in the past, we've used Metabolon, which is a commercial company, and you get a really large set of annotated features from Metabolon. I think they, larger than you can get anywhere else, essentially. But the downside of using Metabolon is they don't give you the raw data, the MS spectra. So it's hard to discover novel things that you didn't know were there. Mm-hmm. And so I think, we've started working a bit with Peter Dorstein. Mm-hmm. So that would be really cool to try to do some deeper de novo annotations of these downstream metabolites that are coming out from degradation of statins. Yeah, it's a good idea.
Cool.
Sean, so we have a question here from Camilo Gomez Garzon that I will rephrase a little bit. So he's asking about whether the same work such as the one you described, was done for type 2 diabetes drugs, such as metformin, that can sometimes cause stomach discomfort, which may come from the microbiome. And also, if you can talk about some of the ways you're taking this work to ways that we can engineer maybe the gut microbiome to alleviate those metabolic side effects.
Oh, great question. Let's see, metformin. Yeah, actually, there's a lot of studies on metformin just because, well, I think it makes sense that it would be associated with the microbiome to some extent because it's involved in metabolism and diabetes and absorbed in the gut. So there are several studies on that. Actually, I know Christian Diener had a study a while back from his prior lab where they were looking at treatment naive, so people not on metformin versus people taking metformin and type 2 diabetes and how that the microbiome was affected by all of that. And I know that there are studies in vitro showing that microbes can eat metformin essentially or degrade it. So for sure, and statins, again, one of the reasons we study statins is we had these big sort of untargeted cohorts that had been recruited. And if you want to find, roll the dice for what drug is the most used in these cohorts that we have the most data for and the most power to investigate, well, statins, right? It's the most used drug in the US. So that was the one that was the easiest to go after. How are we using this and how might we engineer it in the future? So we get some insight for what might be going on the on-target side and the off-target side that could help us with precision drug treatment. So on the on-target side, we find that people who have lower efficacy of a statin have lower LDL lowering, less LDL lowering, so to speak. They are depleted in I think, Bacteroides. And we see a strong association between Bacteroides and blood levels of ursodeoxycholate, which is a bile acid, and it's been associated previously with lowering LDL cholesterol. I think there are some people who take it actually as a way to lower your LDL. There's a cool study from the Sonnenberg lab that showed if you ate L-arabinoxylan, which is a dietary fiber, you could actually boost the level of ursodeoxycholate in your blood. So that could be a means by which you have a co-therapy, where if you're taking a statin, you're not bringing your LDL down as much as you want, you could maybe supplement with arabinoxylan or directly supplement with ursodeoxycholate and try to even further pull down your LDL. On the metabolic perturbation side of things, we notice that people who have the biggest disruption to their metabolism tend to be depleted in butyrate-producing Clostridia and Akkermansia muciniphila. And so there's a reasonable approach there where you could apply probiotics potentially to help shield people from the metabolic effects of the statin. So I know Akkermansia muciniphila now is on the market. I think Pendulum is a company in the US and there's a company in Europe called the Akkermansia company, and you can now buy it and give it to people. So I could imagine having a statin as a baseline treatment and then supplementing it with co-therapies that try to guide the response of an individual person based on their microbiome.
Thank you, Sean. Actually, just a piggyback question to that. So do you see a way one day where engineering the microbiome could directly lead to lowering LDL levels without statins?
I mean, I think a lot of people try lifestyle interventions before going onto a drug. And, one thing that's been done a lot is dietary fiber like arabinoxylan or metamucil, and these things have been shown to bring down your LDL. So you can budget, but maybe you aren't bringing it down enough for your cardiologist to be happy. So I definitely follow your cardiologist's advice there. There's also a supplement out there. It's called like red yeast rice or something, and this is like a yeast that's been fermented with a rice that's been fermented with a yeast. And that fungus actually, if you did metabolomics on it, it would be full of levastatin, right? It's actually I think it's one of the organisms that the drug was originally isolated from, and people take it as like a non-drug therapy, but in a sense, they are taking the drug. Yeah, that's what I'd say.
Awesome. So this is a question from Doug, I think to you, Catherine. Might quorum sensing in bacteria be responsible for some of the inconsistencies in the different roles of Prevotella?
So By quorum sensing, do you mean just?
I think it's communication between.
Yeah. I'm just trying to think, like, I mean, if there's different, there's very different community types, it's sort of like there's just this very dominant, like Prevotella, so numerically dominant, but there is just so many different microbes that correlate, like these, with the Prevotella or with the Bacteroides, in these different contexts. And we did in our paper where we were describing the Prevotella rich microbiomes in MSM, kind of actually do an analysis where we were looking at whether there was differences in co-occurrence patterns in MSM versus not, in terms of, like what other types of bacteria, they correlated within those different, contexts. And there were unique patterns there. So, I can kind of see that there's things that are kind of related to context, on the community level, and how that could definitely be going on.
Actually, can I ask
Yes.
a question?
Go ahead.
There's these papers out of Denmark, I believe, Jorth et al., right, where they show that high Prevotella microbiomes help you tend to be able to lose more weight on a high fiber diet. Have you seen any evidence for that in your MSM cohorts? Like, are these folks more, if they switch up their diets towards a healthier diet, are they better able to lose weight than the average individual?
We are actually, we're doing those studies. So we actually are doing diet interventions. And when we got the grant, we didn't know it was MSM. We were like, okay, we're going to have HIV negative and positive. And that's when we started using the MSM controls. And then we realized, oh shoot, like our test and our control population are both Prevotella rich. So we added a third cohort where we have HIV positive, negative MSM and non-MSM. And we did an intervention. Actually, it was pretty dramatic. Like we gave them fiber, fat, sodium totals that were like the same as an agrarian diet, or we gave them like a Western diet. But our outcome, they were actually non-obese. Our outcome was, we were looking at both metabolic health markers. So like, LDL, HDL, fasting blood glucose, insulin, and those things were actually elevated in our HIV positive, so correctable more so than our HIV negative. And then we were looking at, inflammation, like IL-6 and CRP, and it was part of that whole hypothesis we had of like matching, like, okay, you have a Prevotella rich microbiome, no matter what you eat. So, and a high fiber diet is like especially beneficial if you have a Prevotella rich microbiome. And we were saying in terms of your metabolic health, because there's papers on that with glucose tolerance. So, rather than trying to modify the microbiome, was like, you should eat what your microbiome is like, because, like there's matching that happens in non, and then there is. And so we're still crunching on that data, but, I would say, at this point, I'm not seeing like, we're seeing like a lot of differences in HDL, like with the diet, but it doesn't differ if you're like in one cohort or the other, what I mean? And and the inflammation is like borderline, so it's like almost significant, but not quite. But, but again, we kind of see it in HIV positive, but they're the only ones who are like, elevated to the start. And it's like, they're all Prevotella rich. So it's like, we've been having trouble kind of, seemed like a clever idea, but I like so far it's not like, overly impressed. And like and again, we're still crunching on the data, but the thing we have seen, correlates more with Bacteroides than Prevotella. And it kind of brings up too, when you see these correlates, is it presence of Prevotella or lack of Bacteroides? What I mean? That's really kind of driving what you see, because they do seem to trade off with each other. And even those mouse experiments I showed where they gavage the Prevotella in, the Bacteroides goes down. And yeah, I don't know, they're all relative abundances, so who knows? But it does seem to be, kind of niche competition going there. And yeah, and there's this evidence you add Bacteroides and there's beneficial, influences for obesity and in the context of a high fat diet. So is that what's driving it, or but
yeah, the effect amplifier of either a good or a bad diet, right? It's bad when it's bad, and it's better when it's good. Prevotella.
Yeah. I do think there is I do think, just based on all the literature I've read, that it's like, if you're eating a high fat diet, there's just a lot of evidence to me that Bacteroides are actually kind of more protective in that context. And Prevotella can be detrimental because they can make like TMAO more, that enterotype. But if you're eating a high fiber diet, then the Prevotella shows health benefits. So it's really context specific,
Yeah. The question is it context specific or is it strain specific, and we just don't have the resolution to look at that? So if you had HIV positive individual that consume high fiber diets, what would be the effect of Prevotella then? I don't know if you've looked at that. Could you have then conflicting effects of different strains of Prevotella?
Yeah, and we have definitely not looked at the strain level, although we are at this point, I just have hints of it because when we look at our 16S data, there's a lot of different Prevotella ASVs even, amplicon sequence variants, and we can see like most of them go up in MSM, but there's one that goes down, so like there's some evidence, from that, but we just haven't done the shotgun metagenome with it yet, and we're doing it now, but I think that will be revealing, and it'd be interesting to kind of relate it to the Prevotella coppery clusters that have already been defined, but then see if there's just novel ones too, and whether they actually, the papers done so far, they kind of differentiate across like their carbohydrate utilization genes, but they're looking at people differentiated across diets. In this case, the strains might be differentiated, but I would expect is it more like differentiated along genes that, tolerate inflammation or, or things like that. And, because I don't see any evidence that the Prevotella enterotype in men of sex with men has anything to do with diet. I mean, we've looked at that and, we haven't seen any evidence of diet as a driver. We don't know that.
I think I remember you presenting on this a while back. Didn't you say that some of the MSN actually had higher meat diets?
They actually, on average, did. Yeah, so it's like the opposite.
I have another futuristic question for you, Dr. Luzapon, from an anonymous participant. Do you think that your studies can help prevent HIV transmission or infection? And if yes, how could the microbiome modifications be a strategy?
Yeah, I mean, in theory, yes, because, we have evidence that the microbiome of MSM is influencing the expression of HIV co-receptor CCR5, and CCR5, I've already heard that's the one that the couple people have been cured of HIV was they did a stem cell transplant and they put in CD4 T cells that didn't have CCR5. It's like essential, and if the microbiome is upping the CCR5, then modifying it, in theory could help prevent it. So but then that gets down to the problem in every study. How do you modify a microbiome? I mean, I think I think there's one of the things is just understanding what drives that microbiome type is important, because then you could, go after the drivers. But if you're just going to say this is the microbiome type and we want to prevent it, I really think that's why we're trying to get more at the mechanism, like, with taking out particular microbes and looking at strain level variation. I'd love to just kind of better understand it. Well, it's this compound that this microbe makes that's interacting, with this, that's inducing the CCR5, and then you target not a microbe, which right now with any disease you just kill everything, right? Like antibiotics, I think you can more target, pathways, I like the type of work I've seen Stan Hazen go at TMAO. We're going to just try to make the microbes, not make it. We're not going to try, but you need that mechanistic understanding to do that sort of thing. So.
Sean, that's a question from Richard Sprague, and he's asking how much diet information did you have about statin users? Could it be that some of them have had low carb diet that increased LDL level, and did you take into account that?
Good question. We really didn't have much dietary information, sadly, for this particular cohort. We did our best to control for as many covariates as we could, so we of course controlled for things like sex and age, BMI. For a lot of our analyses, we were even controlling for things like type 2 diabetes and in other drug usage. But yeah, we don't quite know what the dietary intake is for those folks. You can guess that people who are in like the BAC2 enterotype, for example, in prior studies, it's been shown that those folks are often the people who are on lower fiber diets. So you might be able to infer something about diet based on composition, but technically we didn't have that data for that data set. Although, let's see, we had a validation data set from Europe, Metacartis, and we did not have data there.
We didn't have
we didn't have diet data. We have metagenomics though, and we've thought a little bit about trying to infer some dietary information from metagenomes because there's some amount of DNA from plants and animals in our poop. It's like 3 to 4% of all the DNA is from non-human plants and animals, as we've looked at so far. Christian has been working on this a little bit. So we don't know if that's a feasible way to get unbiased diet estimates from metagenomics, but we're kind of working on that right now.
Okay. And I have a question for Dr. Luzapon, and help me here, Sean. Enosh Kazam is asking for the carbohydrate degrading enzymes measurement in Prevotella capri, and in general, in addition to metagenomic data that's only looking at the genome, what are your thoughts on the transcription or translation of these genes, and is there a needed time frame for the bacteria to acclimate to the utilization of complex carbohydrates in the diet?
That's interesting questions, and yeah, I think transcriptomics, I think in general is kind of underutilized in the microbiome field in general, and so yeah, I really don't, I don't have a good sense of, transcriptionally. I think that would, just be faster, that organisms could quickly turn on genes, just knowing how organisms act, to, utilize a new diet source, but I have seen evidence, in the literature just of it taking some time for the right microbes, to just be the right microbes to get there to utilize a new dietary substrate. I mean, you just see, I've just seen interesting papers with things like Japanese populations more likely have microbes that degrade seaweed, or or soy, they say soy compounds are anti-cancer, but it's actually microbial metabolites of them. And you actually see that effect more in populations that eat a lot of soy. So, I think if you never ever eat fiber and then you suddenly do, it's unless you have microbes there that could quickly change their transcription, it's going to take a little time to be able to fully utilize that, or the right exposures, because I don't know, to the degree to which useless microbes are left around. Even the talks we heard this morning about, the loss of these microbes in agrarian cultures in the Western, I don't think they really definitely get kept around, and there's ones we're losing, permanently. And so I think that's my bigger concern. Just, you don't have the microbes at all. And I think if you have them, they could probably turn on the right genes pretty quickly, but that's just my impression.
Yeah.
Just a final question to either of you. Javad, he's asking about how the microbiome, you discussed different things that can change the microbiome, And the question is, to what level the microbiome is robust or resilient to all these factors, drug, sexual intercourse, or are there different compositions that are more resilient to your knowledge?
It's good. It's a good and hard question.
Yeah.
Kathy, do you want to take a first stab? Or?
I could take a first stab. I mean, I think, I think resiliency and what the first thing that kind of comes to me is my thought are just things like, does redundancy and high diversity and things like that promote, resiliency to disturbance. And actually one thing, kind of, from my own work actually that I could think of is like, we actually work in some mouse models of C. diff, and, we find that like when we feed, it's an antibiotic model, and there's been papers showing this. If you like feed your microbiome fiber, it's more resilient to the antibiotics, right? So it's sort of like, I think, a healthier microbiome that's getting everything it needs, all the food, so it can maintain diversity and maintain redundancy, is going to be, more resilient to stresses. So I think that's one of the major things I kind of think of with that. But what do you think, Sean?
Oh, yeah, I'd agree. I think diversity can promote resilience, right? The microbiome is constantly getting whacked around by, one day you're eating bananas, the next day you're eating tangerines, and different things are flying out at different times. And so the more diversity you have of organisms that kind of can respond to that variation, the more stable the system tends to be. Although some of our work has shown that you definitely see people with very high diversity microbiomes it isn't always a great thing, right? Constipation is actually associated with high diversity. And maybe constipation is resilient, I don't know, but it's not necessarily a good state you want to be in. In my postdoc, I did some time series analysis work, and there we see pretty strong stability, at least in terms of people who aren't experiencing any major lifestyle changes, aren't flipping their diets in a radical way, or aren't taking antibiotics. In that such situation, on month to year timescales, the microbiome looks very stable. Like the average abundance of all the taxa, even though there's fluctuations day to day, remains pretty stable. And maybe on decadal timescales, there's like a slower drift, but from like months to years, it seems pretty stable over time.
I've seen that too in our work. They kind of bop around their own little mean, it's not, yeah. In fact, in some of that, we just use that to discount like time between samples, even as a factor, because it's like, there's not a directed change, like in the year or so time scale you're looking.
Nice.
Okay, we are out of time, and I apologize for those we've not been able to get to their question. So I'd like to conclude this Q&A section and thank Dr. Gibbons and Dr. Luzapon for their fascinating talk and this engaging Q&A session. So this is the end of this session. I'm hoping to see you at 12:45 in 15 minutes for session 3 that will focus on social determinants of healthy gut. Thank you.
Thanks. Bye, Kathy. And sorry you missed Dr. Jimenez, everybody. That's too bad. I hope she's okay. But yeah.
Bye.
Good job stepping in last minute talk. That was good.
All right. So welcome back to the third and final session of our symposium, where we will be discussing social determinants of a healthy gut. I am thrilled and honored to introduce Dr. Amato, joining us virtually from Chicago, Illinois, and Dr. Benezra, joining virtually from Stevens Institute of Technology in Hoboken, New Jersey. Please continue to leave your questions and upvote the questions of others in the Q&A function for discussion during our session after our two 30-minute talks like the other sessions. All right, so our very first speaker, Dr. Catherine Amato, is an associate professor in the Department of Anthropology of Northwestern University. A trained field primatologist, she is president of the Midwest Primate Interest Group, editorial board member of Folia Primatologica, and author of over 75 publications focused on gut microbiota, nutrition, primate ecology, and how these inform human adaptation around the world and evolution throughout time. In addition, she is a CFAR fellow in the Human and Microbiome program, where she has also been recognized for her work in global health. She will talk with us today about the human gut microbiome and health inequalities. Welcome.
Thank you so much. It's a pleasure to be here. So yes, today I'm going to talk a little bit about my work in the area of the human microbiome and health inequalities or inequities. This is really kind of a call to action or a kind of drawing of attention toward what I see as an important gap in the current field of microbiome science, particularly host-associated microbiomes. And so let me go ahead and jump in here. So we know that there are health inequities in all sorts of health outcomes, and these are often linked to socioeconomic status. So for example, in the US, men in the highest and lowest percentile of income have an approximately 15-year difference in life expectancy. So there are certainly differences in health outcomes in certain groups of people. And this is often linked to socioeconomic status, as I've just pointed out. But in addition to socioeconomic status, things like self-identified race, sexual identity, and gender status can also have powerful impacts or be powerful predictors of many health outcomes. So for example, if we control for SES, Black adults in the US have triple the odds of being diagnosed with diabetes compared to white adults. And LGBTQ adults are twice as likely to report multiple risks for cardiovascular disease than heterosexual individuals. Now, often these inequities in health outcomes that map on to race or sexual and gender identity are kind of implied to have some sort of biological underpinning. But I want to be particularly clear in the case of race that we're not actually.
I hate to interrupt, but your slides are not quite sharing yet.
Oh, sorry.
The share button is?
Yes. Hang on. Okay.
We can see them now. Thank you.
Excellent. You're welcome. All right. Apologies for that. I had it all queued up, but not shared. So you didn't really miss anything in terms of graphics. I just wanted to point out that when we're talking about racial groups, oftentimes it's implied, especially in biomedical research, that there's some sort of biological underpinning to any differences in health outcomes that we're seeing as a result of race. But when we're talking about race, ethnicity, ancestry, and genetics, we're often talking about very different things. And race, in fact, does not have a biological underpinning that could be kind of de facto explaining these differences in health outcomes that we see. We know that biology adapts to local environments. So there are genetic differences in populations that live in different geographic areas, but these are very locally based differences, right? Whereas categories such as race and sometimes ethnicity are usually talking about larger groups of people that can be identified by, let's say, kind of visual characteristics, right? So for example, if you think about Asian American, right, that's a really broad category that doesn't say a lot about people-specific geographic kind of ancestry. So there are groups of people that are from different countries, and within those different countries, different populations in different parts of the countries may have local adaptations to certain environments. But when we group all of those people together, we wouldn't expect there to be specific biological differences within that group compared to other people. It's just kind of too big of a category. So oftentimes, while we have these kind of implications that, you know, perhaps these racial differences are because of biological differences, there's really no genetic evidence that race is a biological category. We do know, however, that we see these differences in health outcomes as a result of race or associated with race and other kind of groups. And so what I really want to emphasize is that race is a social construct. And what we're seeing when we're seeing differences in health outcomes with respect to race is that we're seeing these being kind of impacts of the social environment on biology, right? So the types of environments that people live in and their day-to-day experiences are going to affect their biology, and those do differ by race, particularly in places like the United States. Now, experiences with discrimination as people associated with certain social groups can be personal, right? So people can experience direct experiences of discrimination, right? Somebody has an actual negative event happen directly to them. But these can also be structural, right? In terms of how resources are allocated, laws and policies that determine who gets access to resources, whether those be financial resources or other sorts of environmental structural resources. Those often also have biases for or against certain groups that can change the environments people are living in, which can then impact their biology and ultimately their health. So these racial differences in health outcomes are a real biological phenomenon, but they're not because of underlying biological differences, and instead are a result of how the environment that certain groups of people are in many cases forced to live in are going to affect their biology and their health. Now, there's been a lot of research about how this may actually kind of work in terms of mechanism. And so one example is that there's been a lot of interest in the HPA axis, so essentially understanding how stress affects biology. And often this is through interactions with the brain and the endocrine system, and thinking about how that could activate or affect the immune system and lead to inflammation, and then prolonged exposure to inflammation can then alter biology potentially in negative ways, right? So stress goes to inflammation, then there's downstream effects on a multitude of body systems. There's likely other pathways in addition to this one, although this has been a very powerful explanatory pathway. And today I'd like to point out that we should be thinking about the microbiome as another one of these potential pathways. And I'll add a pathway that's probably interacting with this inflammation pathway as well. So essentially the idea here is that we may have the environment, which can affect the microbiome, which can then feed back and affect host physiology. And if you're having this happen, you know, there's differences in environments that people are living in, that can mean that they have different microbiomes and different impacts on their physiology. Now, kind of from a baseline here, I want to remind those of you that don't know that the gut microbiome can be influenced by many environmental factors that their hosts experience. So diet, what people are eating, can affect the microbiome. We have things like fiber intake thought to be a beneficial impact on the gut microbiome, in that you have microbes that can degrade that fiber for us and produce compounds that can be used as energy and are also signaling molecules in many different body systems. Daily practices such as the medications we take can alter the microbiome, as well as our social networks, right? Who are we coming into contact? How many different people are we coming into contact with? And what types of contact are we coming into with those people, right? Are we shaking hands? Are we living with them? Are we sharing office space? All of those things can affect the microbiome. We also know that the gut microbiome can feed back and affect multiple body systems. So the microbiome can affect nutrition. Like I said, it can produce molecules that can serve as an energy source for hosts. It can also produce vitamins and break down toxins. It's been shown to train the immune system early in life and can also affect immune system later in life, immune system function later in life. And it also looks like it's training the nervous system and affecting its function both in early life and later life. And it's been associated with variation in behaviors like risk-taking in some cases, and associated with things like anxiety and depression. So given this kind of foundation, it makes sense to think about how inequities in environments associated with groups that are often discriminated against could be associated with differences in microbiome exposure and composition, which then could lead to some of these health inequities that we see, right? So specifically, if we look at how environments differ at individual, household, neighborhood, regional, or national and global levels, what does that do to things like dietary quality, stress, access to outdoor spaces, all of these kind of different environmental factors that could be affected, which could then feed back to the microbiome and affect biology. But this is kind of an open question. What are kind of the pieces of evidence that we have out there to address this? So I want to go through some examples kind of briefly here. I want to start by giving you some evidence that structural inequities could be affecting microbiome composition. Before I dive into that, I want to briefly talk about what a healthy microbiome is. This is a huge question that we don't have a good answer to yet, but kind of the two things we like to think about more generally are diversity of the microbiome. So we tend to think that more different types of microbes in the gut, in this case is what I'll be talking about, are better because if you lose some of them, some others might step in and fill that function, or there's kind of less resources for an invader like a pathogen to come in and kind of take hold. That's not true in all cases, but often more microbes are thought to be better. Also in terms of thinking about community function. So what microbes are there and how are they interacting, right? Is there one key kind of microbe that's providing a lot of functions, or are there certain kind of functional groups of microbes that are important? So it's really kind of thinking about both diversity and kind of the composition and function of this community. So in terms of thinking about how environmental inequities could be affecting the microbiome, I want to start in early life. Specifically, I want to talk about C-sections, breastfeeding, and essentially skin-to-skin contact and kind of direct microbial transfer through social contact. These are things that we know we see disparities in terms of that there are populations that tend to have more C-sections because of biases in our medical system, as well as perhaps pre-existing conditions that are a result of underlying inequities also. Things like breastfeeding and skin-to-skin contact, there's inequities in there. In terms of who has access to parental leave, what are the kind of accommodations that are given in the workplace in terms of things like pumping, etc. These are things that certain groups of people have more or less access to than other groups, and they often fall on lines in terms of minoritized populations, right? Many minoritized populations tend to have higher incidence of C-section, lower ability to breastfeed because of social structures, as well as lower ability to do skin-to-skin contact. And with all of these, we see differences in the microbial community associated with them. So if in this first study on the bottom left hand, you can see that babies born via C-section, which are in the light and in the blue color, light blue dots. These graphs, so I'll say each point represents the overall microbiome fingerprint of an individual, and the closer together points are, the more similar those microbiomes are. So light blue dots are babies that were born via C-section, and light red or the pink dots are babies that were born vaginally. And you can see that their microbiomes look very different at birth. Specifically, babies born via C-section tend to have microbiomes more like mom's skin, and babies born vaginally have microbiomes more like mom's vaginal tract. We also see this interacting with breastfeeding in the next kind of middle plot here. On the left, you have babies that are not exclusively breastfed at 3 months, and on the right, there's exclusively breastfed babies. And then within that, the first two graphs are looking at vaginal birth, and the next two are C-section. And you can just see these are relative abundances of different microbial taxa. And just by looking at the colors, you can see that there's differences based both on C-section and breastfeeding. And then finally, the last study on the right, this is actually from non-human primates, where we have really detailed data on social interactions and how often individuals are spent grooming, which is direct physical contact. These are baboons. And the more time that you see individuals spending grooming together, which is as you move right on the x-axis, the less dissimilar their microbiomes become, which is on the y-axis. So essentially they're becoming more similar to each other. There's less dissimilarity, which again is indicating that, there's an influence of contact in terms of the types of microbes that are being transmitted between individuals. So if you're having differences in breastfeeding, C-section, and skin-to-skin contact, this is going to be affecting the types of microbes that infants are exposed to in early life, and essentially setting them up for potentially different microbial composition. Later in life, there's a couple other factors I just want to give you a few examples on. We know that diet can affect the microbiome throughout life. And again, we tend to see disparities in the types of food that people have access to. Particularly in food deserts, we tend to see people having more access to processed foods, fast food places, and less access to fresh produce. This is a study where they changed the diet dramatically over a period of 4 days in a lab group, where some people went to a completely plant-based diet and some people went to a completely animal-based diet. And basically the change in the slope of the line is how much the microbiome changed kind of overall. And you can see that as people move to this animal-based diet, which was basically a meat and cheese diet, so more processed food, less fiber, there's substantial changes in the microbiome. So again, you can imagine that people that are living in food deserts and don't have access to high fiber foods are going to have a very different microbiome compared to people that do have access to those foods. Environmental exposure is another important thing to think about. So we have whether or not people have access to safe green spaces, right? This is an issue in a lot of urban environments where there's just not safe places for people to spend time outside. And yet we know that spending time outside can affect the microbiome. So this was a study actually in a daycare situation where they had a standard daycare and a nature-based daycare. And then there was an intervention where they essentially brought dirt to the standard daycare. And you can look at the diversity of the microbiome. That's what these bars are showing. And you can see that after dirt was brought into the standard daycare, the diversity of the microbiome actually increased in that kind of first pair of bars, and actually went up to the same level as the kids that were already in the nature-based daycare. So there's this basically influx of microbes that kids are getting from having exposure to the natural environment. And then the last thing I want to talk about briefly, this is actually from a mouse study, but this is evidence that suggests that circadian dysregulation can affect the microbiome. So these are mice in a lab. They're all being kept in the same conditions, except that there was a shift in essentially when the mice had daylight, right? So the circadian dysregulation. And again, we're looking at diversity here, and you can see circadian dysregulation on the right. Those mice had lower microbial diversity than mice that were not exposed to that circadian dysregulation. So again, if we're thinking about certain populations that are more likely to be working in kind of shift work and be more have more circadian dysregulation because of structural forces that are leading to people having to be in those jobs, that's also going to affect what the microbiome looks like in terms of composition. All right, so that's all well and good in terms of trying to think about how differences in the environment could be affecting the microbiome in the context of kind of disparities and inequities. But does that mean that these differences are actually going to affect health outcomes? Well, we can look at some of the data that's looking more directly at health outcomes and microbiome variation, although it's not taking into account the environment and kind of where that microbiome variation is coming from. So I'll give you a few examples here. The first is looking at metabolic disease, in this case, obesity. And on each of these slides, I'll show you data on the left that's observational from humans, and then data on the right that's coming from mouse studies that's supposed to get more towards causation, because the real differences in these mice are the microbes they have and not necessarily, environmental differences. So you can see here that people that are considered lean have a higher microbial diversity than people that are considered obese. And when we take those microbes and put them into germ-free mice, these are mice that don't have any microbes of their own, we actually can see those phenotypes replicated. So mice that get microbes from lean human donors have essentially no percent change in fat mass, while mice that get microbes from obese human donors have an increase in fat mass. If we look at similar data in relation to asthma, on the left hand, these are data showing the difference in microbial composition. So each color is a different tax of microbe. And you can see in kids that were controls, kids that have atopy plus wheeze, and then each of those symptoms on their own. And you can very clearly see that there are differences in the types of microbial taxa that those kids have, depending on what sorts of symptoms they have. When we take those microbial communities and we put them into, again, germ-free mice with no microbes of their own, and then look at inflammation scores that are associated with things like asthma, we actually do see differences. So AW stands for atopic wheeze, and naive are mice that got they didn't get microbes from kids that have wheezing, essentially. And so you can see they have a much lower inflammation score than that kind of middle bar, where they got microbes from kids that do have a wheeze. The last treatment was actually when they put back in microbes they thought might be probiotic and kind of help ameliorate this. And you can actually see a slight decrease in inflammation when those microbes are put into the mice, in addition to the microbes from the kids with wheezing. And then finally, There is also some evidence that we see microbial community composition and function interacting with depression and potentially playing a causative role. So on the left, these are essentially, this is a graph that's showing that certain microbes are associated with major depressive disorder. These are the ones in the kind of red bars that are going to the left, versus healthy controls and the green bars that are going to the right on the top. So essentially you have different microbes characterizing people with those different kind of clinical evaluations. Again, if we take microbes from people with these clinical evaluations and put them into germ-free mice, what do we see? Well, we see when these mice are put into an elevated maze test or an open field test, so this basically looks at how willing the mice are to go out and kind of explore a new area, we actually see the mice doing less of that when they get the microbiomes from people that have been diagnosed with major depressive disorder compared to mice that get microbiomes from healthy controls. There's some, we could debate about whether that's actually kind of a symptom of depression in mice or not, but nonetheless, it's an interesting behavioral pattern to think about in the context of these data. So overall, I would argue that it seems like the literature overall suggests that there should be these connections. We should expect these connections to exist between looking at environmental disparities, kind of structural discrimination, how that affects environments in the middle there in the pink, and then how that feeds back to affect the microbiome and ultimately health outcomes. So what do we know about this in the literature so far? The answer is really not a whole lot. There hasn't been a lot of work that's really linking all of these kind of points together. This is a table that I put together in 2021, looking at all the studies that kind of focus on SES as one of their main outcome variables. There's not very many of them, and they're also picking really specific measures of SES, and they're not a lot across a whole lot of contexts. I looked earlier this morning, and there's like a handful more studies than there were when I put this table together, but not very many. Here's what we have so far in race and ethnicity. Also not very much, and I also want to put the asterisks in here that race and ethnicity are getting used in very different ways in these studies, and often not in inappropriate ways, that in terms of kind of them being social groups. But there are some of these studies, and there has not been a whole lot on this that has come out since then. I know people are working on this, but there's not a lot in the published literature right now. And then in terms of looking at sexual and gender identity, there's really nothing out there in terms of thinking about structural discrimination and kind of the social implications of the environment on health. There are studies that are looking at men that have sex with men and how that affects the microbiome of like the genital tract and kind of disease risk, particularly with respect to HIV, but nothing that kind of goes beyond that. So based on all of this, I would argue that what we need to do moving forward, assuming that we believe there are these links between structural discrimination, the environment, the microbiome, and health, is really start to get at this in more detail. And so some of the things I would suggest first and foremost is that we need to operationalize and quantify racism and discrimination, right? Instead of just throwing race in as a variable, actually say what we mean about race and measure that, right? I think we also need to explore multiple levels of inequality and their relative importance or interaction, right? So when we're talking about inequality, are we talking about things at the household level? Are we talking about things at the national level? Is one more important than the other? And like I said, do those interact with each other? We also need to empirically link the social environmental factors to the microbiome to the health outcomes in a single study. Right now, we have the environment to microbiome link in many studies and the microbiome to health outcomes in many studies. But there aren't studies that I know of that link all of those together in the published literature right now. I think we need to better establish mechanisms and causality and elucidate the relative importance of the microbiome versus other pathways, right? Even though I'm arguing that I think the microbiome is a really important pathway by which some of this may occur, it's certainly not the only one. And so how important is it relative to potential other pathways? So to do all of this, I want to emphasize that we really need comprehensive data from interdisciplinary studies, right? There are things that people with a biomedical background are going to be much better at designing for a study versus people with a social science background, right? Maybe that's in terms of kind of operationalizing ideas of racism and structural discrimination, or thinking about methods for maybe epidemiology to look at these things at a broader scale and get at causation better, versus trying to get better taxonomic resolution and trying to understand what function versus taxonomy means in terms of the microbes. And along with that, we have to generate these data in a way that makes it actionable, right? We need to make sure that the people that are designing the studies have the training they need to be able to design the studies effectively, and also that the people that are being included as participants are representative of the populations that we think would be most benefited from the types of outcomes that we're getting from this, right? If we're doing all of our research on predominantly white populations, how do we know that's going to translate to some of these minoritized populations that may actually have kind of these disparities in health outcomes and could maybe benefit more in some ways from some of the potential interventions that are being developed? The other thing I want to mention is kind of how are we going to intervene when we talk about interventions? Kind of, what do we do next? And I think there's kind of two different directions one could go here, and I don't think they're necessarily exclusive. But kind of the two areas I want to talk briefly about as I wrap up here is thinking about targeted biomedical interventions, so kind of personalized medicine types of technology advances versus ecologically inspired interventions. And these are things that I think of as more like preventative and kind of policy-based, right? And breastfeeding is a great example. So we know that bifidobacterium is a really important microbe that's passed to infants during breastfeeding. And so there's huge potential there to kind of focus on this microbe as a key player and develop like special probiotics for this type of microbe, right? That can actually be delivered to infants that are lacking it. But at the same time, it would also be useful if we could look at policies surrounding parental leave and breastfeeding so that we have infants that are more able to engage in these behaviors and might not need that targeted biomedical intervention, right? It's really important to think that when we're developing these kind of fancy new biomedical intervention, who's going to have access to that, right? How much is it going to cost? Is it going to be distributed through these medical channels that may already have biases in terms of who they serve? So they may be very useful, but we also need to think about policy shifts that will allow us to kind of help people in ways outside of the traditional medical system. And so just as kind of a final thought here, in terms of thinking about how we can start to kind of better integrate policy and research, is that there's many layers here, right? That the policy and the research should really be interacting with each other in terms of thinking about things like, federal funding agencies. Are they putting in requirements about how studies are designed and what kinds of populations that they're targeting, right? And in terms of medical associations having certain standards, et cetera. And then also looking at, where funding is coming from and how people are selecting populations for their study at kind of a more local level before we can get towards these really kind of more specific interventions. So there really needs to be this back and forth. I think that can be hard to do, and I think there's efforts being made at it, but I think that we really need to do a better job of more fully integrating these discussions so that we can get to outcomes that are actually helpful for people. So with that, I'd like to thank the Human Capital and Economic Opportunity Working Group, which is kind of the organization that helps spur some of these ideas, as well as the co-authors on the paper that we kind of put together as a result of this workshop. A lot of different microbiome scientists that we're kind of trying to think about, how can we bring together what we're all have expertise in on kind of the science medical research side and think about how to better integrate with this in the context of health inequities. And that's all I have. Thank you.
Thank you very much, Dr. Amato. That was a great talk. We have some good questions in the QA that we'll get to at the end of the session. And we'll move next to Dr. Benezra. So Dr. Benezra, as I go through your introduction, go ahead and start sharing your slides. I know it always takes a couple minutes to set that up. So our second speaker, Dr. Amber Benezra, is an assistant professor of science and technology studies at Stevens Institute of Technology. As a sociocultural anthropologist, Dr. Benezra's academic focus lies at the intersection of anthropology and microbiology. Her work, based on field studies in Bangladesh titled Biota, Co-Evolving an Anthropology of Microbes, is the very first ethnography of the microbiome. She is a pioneer in the exploration of how combining microbial ecology and social science could benefit global health. And she will talk to us today about who decides what healthy is, studying microbiomes across disciplines.
Thank you so much. Are my slides good?
Yeah, you were great. Thank you.
Great. All right. Thank you so much for that introduction. I'm so happy to be following Dr. D'Amato because her work is wonderful and a perfect segue to what I want to talk about. And thank you to Sean and Catherine for inviting me to be part of this symposium. This talk is going to be much different than most of the talks that you've heard today. Well, no, it's going to absolutely be totally different than everything you've heard today because I'm a cultural anthropologist by training. That's what my PhD is in. I'm really glad to be here though. And I think it says something very significant that I'm a part of this symposium and I'm really grateful for that. So hopefully this will all make sense to you all. And anthropologists typically read their papers. And so that's what I'm going to do. I'm going to be reading. So hopefully that's not too jarring. It's very different than a scientific presentation of data and how scientists usually talk about their work. But here we go. Okay. Dhaka, Bangladesh looks lovely in the earliest hours of the day. When the sun rises over the bamboo scaffolding of half-finished high-rises, the people are quiet and birds and insects fill the sky. The morning light is beautifully tinged with exhaust, pollution, and dust. Within minutes, it becomes intense, bright and hot, the UV index often forecasted at a maximum 10. The sun starts early here. In Mirpur, one of the most densely populated localities and the most densely populated place on Earth, mothers rise in one-room shacks, chase mice from cooking pots, boil water, and prepare food for the day. 8,000 miles and 11 hours later in St. Louis, Missouri, scientists will arrive at the Gordon Lab, sacrifice mice, segment intestines, and prepare cells for sequencing. The microbes in the guts of these mice are from the guts of Bengali babies. They traveled across the world in test tubes, in fecal samples collected by mothers and packed in picnic coolers in the stifling Dhaka sun. These microbes traveled by airplane on flights that cost more than the yearly income of most Bangladeshi families. They arrive at the Center for Genome Sciences and Systems Biology at Washington University, a gleaming multi-million-dollar facility with several sequencers, a mass spectrometer, and a computer cluster capable of analyzing trillions of reads. These microbes will be injected down the throats of black 6 mice. I think many people were talking about germ-free mice in their studies, so that's what the Gordon Lab has as well. There's more here than a predictable cross-cultural comparison between a globally southern slum and a globally northern scientific laboratory. These spaces are more than geography, more than poverty, and more than notions of power, histories of colonization, more than practices of living and knowing. They don't just overlap at the boundaries of the body. Between the Gordon Lab and Mirpur, the social and biological become equally important to understanding human microbial relationships. Based on a collaborative partnership with scientists working on the microbiome, my research follows commensal microbes in the human gut as objects around which scientific and anthropological imperatives are emerging. I conducted fieldwork at the Gordon Lab at Washington University School of Medicine, where scientists were studying the interrelationships between diet and microbial community structure and function, and whether differences in human gut microbial ecology affect predisposition to malnutrition in children. I also worked at the lab's field site in Dhaka, where Bangladeshi mothers and children were enrolled in a microbiome and malnutrition study. There I worked with scientists and researchers at the International Center for Diarrheal Disease Research, known as ICDDRB. The Gordon Lab was attempting to develop a translational research pipeline in order to alter the microbiota of those suffering from metabolic disorders and nutritional deficiencies. I followed microbes to homes in Dhaka, where microbes were conceptually non-existent and practically a matter of life and death. As lab members studied the relationships between gut microbes and malnutrition, I explored how an anthropologist could become a member of a scientific team and contribute ethnographic data, while asking biological scientists to make space in research design, experimentation, and implementation for social science expertise and thinking. The sort of whole idea was to make the ethical, legal, and social implications of the science not an afterthought to the scientific work, but part of the research design. In our perspective piece in PNAS in 2012, Dr. Gordon, Joe DiStefano, and I created what we called an anthropology of microbes. We wrote, quote, "The development of new methods for understanding the microbial world provides an opportunity to reevaluate the way that we view our human, biological, and cultural diversity," end quote. We began to consider how the natural and social sciences might use studies of the microbiome to jointly engage pressing public health problems like malnutrition. Sociocultural anthropologists study the complex cultural lives of people, their decisions, their social practices, and relationships. We use the methodology of ethnography to ask questions about people in the world. We do what we call fieldwork. The ethno part is observing and talking to people who are going about their everyday lives as they experience the commonplace and the extraordinary. The graphy part is when we take all that information and we analyze it and write about it. That's me on the right doing ethnography, and my fieldworking partner, Joe DiStefano, on the left. Our work is primarily qualitative. We don't produce clean, noise-free data, and our sample sizes are usually tiny. For instance, I talked to 9 families during my fieldwork in Bangladesh. It's a very small N. Sociocultural anthropologists don't like to make generalized conclusions about cultures, societies, or groups of people, but ethnography provides insights into what life is actually like for people on the ground, giving important details that might not be visible in microbial genomics, for example. It can shed light on the transnational and local realities of people who are embedded in global scientific research and health programs. It can show why health interventions don't work, why they aren't practical or implementable in real life. As an anthropologist, what I love about the microbiome is that it brings together social intimacies with our biological selves, as Dr. D'Amato was talking about. All of the things about who we live with, where we live, what we eat, all determine what our microbial populations are. They're affected by how and where we're born, what food we eat, who we live with, who we love, and those microbes are digesting our food, training our immune systems, interacting with our states of health and illness, moods, and behaviors. We know that new metagenomic technologies are revealing the impact microbial communities have on human health and are changing ideas about biology and what the human even is. In turn, microbial concerns are emerging for the social sciences, humanities, and philosophy. In my subsequent research, I've continued to develop an anthropology of microbes, drawing together the collaborative capacities of ethnography to actively contribute to scientific projects, partnering with scientists to help develop more holistic, analytical, and methodological frameworks with the goal of understanding the embodiments of biology within social dynamics. These transdisciplinary collaborations are not easy, even if both parties are in the partnership come to the table with open minds, willingness, and generosity. We all know that each of our own disciplines has a particular language, a specific way of conducting research, and even a special way of writing papers. We never really learn how to talk to other kinds of scholars or how to work together. I'll say, just as an aside, Dr. Gordon was probably the most magnanimous and generous collaborator I could have hoped for, and still, in the end, our collaboration did not succeed. I worked collaboratively with the Gordon Lab for about 7 years. For 14 months, I was actually in the lab, conducting my independent research and also serving as an anthropologist in the employ of the lab. I was thinking about how accounting for microbes changes anthropology and how anthropology can collaborate with scientists accounting for microbes. The Gordon Lab scientists sometimes talked about humans as superorganisms, a composite of human and microbial cells, an amalgamation of human and microbial attributes. So I'm sure you all know all of these statistics. I know that more recent data shows that there are 46 million microbial genes in the gut alone. Early estimates said 2 million microbial genes in humans. Obviously, this is really interesting for an anthropologist to think about what humans are if we're actually made up of all these different organisms. At the Gordon Lab, a lot of the Gordon Lab experiments were looking at the gut microbes of babies in Bangladesh and Malawi when I was there, who were suffering from malnutrition, trying to figure out the connections between bodily bacteria, food, and illness. They enrolled mothers and babies in the Microbiome Discovery Project, gave them free healthcare and medicine, and took periodic samples of blood, urine, and lots and lots and lots and lots of feces. Meanwhile, I spoke to these mothers, the Bengali field researchers and scientists, and explored ways to reconcile the scale differences between the lab, the intimate biosocial practices of Bangladeshis, and the looming structural violence of poverty, poor sanitation, and infrastructure. I also talked to the Gordon Lab PhD students, postdocs, and technicians to find out how they were thinking about the work they did, as well as their own relationships to their microbiota. So I asked then, and I continue to ask, what's at stake within these newly understood human microbial relationships? How can we address the bioethical issues? Who owns your microbes? Why are certain bodies, especially those from historically vulnerable populations, the focus sometimes of microbiota sampling? What does it do when science talks uncritically of people of different races having different microbiomes? How can we make sure the study subjects are heard and benefit from the research? How does our genomic intertwining with our microbial partners change how we understand relatedness or selfhood? What does it mean to define our states of health through our microbial selves? When thinking about this symposium, and especially this session topic, I realized that social and biological scientists may not even start with the same idea of social determinants or even health. For human microbial ecologists, the health that is important to studying the microbiome might consist of diet, geography, genes, mode of birth, vaccinations. As an anthropologist thinking about mothers and babies in Dhaka, all of those things matter, along with important influences like gender, class and ethnicity, working in environmental conditions like garment factories that exist in Dhaka, the violence of a colonial national history, corrupt or non-existent governmental systems of sanitation and water. We see that when we start to pile up all these concepts of health on top of each other, the picture becomes very messy and confusing. But it also becomes more detailed, more complete, and eventually, hopefully, more precise. This is why microbiome science and anthropology need each other. While I was in the Gordon Lab in 2010 and 2011, at least 6 lab members were working specifically on the gut microbes and malnutrition, and 3 were dealing directly with samples from Dhaka. The number of lab members, as well as those working on both malnutrition in Bangladesh, has substantially increased in subsequent years, becoming a central focus of the lab's work. My research focused on 2 projects, one on analyzing the Dhaka samples and the other conducting diet experiments using what they called Bangladeshi food and germ-free mice humanized with microbiota from Bangladeshi donors. I was specifically interested in these studies because they analyzed microbial material from babies in a specific geographic locale with a specifically defined health and nutritional status. These studies involved human beings I would be able to talk to at a node in the network I could travel to and observe. I wanted to find out where the feces came from, what the conditions were in the place they were produced, and for the people producing them. So this is one of the study families, 2 of the children that were, the baby was enrolled in the study. I just, as an aside, want to say, as I always try to point out when I present these slides, I'm really reluctant to share photos of the Bangladeshi people I took during my fieldwork. On one hand, I think it's really, really important that all of you, and especially biological scientists, see faces of human beings. Often we think of scientific study subjects as samples, right? Fecal samples, metagenomic reads, the data that their bodies produce. So it's really important to see the human being. On the other hand, there's a lot of race, class, and national privilege that I have in portraying people this way. And I really am resistant to be a part of any kind of exploitation of those people. So I still haven't figured it out. So I will show some pictures, but I don't feel comfortable about it. I was interested in how Bangladeshi diets were represented in mouse experiments and how dynamic human eating would work its way into the scientific practice or the ways it would be left out. For instance, Bangladeshi food has a lot of chilies, garlic, and other spices in it. Those things can't be given to mice, they will die. So the sorts of diets were very edited, right, in terms of representing what a Bangladeshi diet actually was. The birth cohort of Mirpur consisted of children whose mothers were enrolled while they were pregnant. These mothers received extensive prenatal care, education on healthy eating, breastfeeding, hygiene, and gave birth at health centers rather than at home, which is the local convention. The case control children were given macro and micronutrient supplements as well as therapeutic foods. All the families received free healthcare in exchange for their participation. This also is complicated and not value-free. The families I spoke to often felt like they were getting crappy healthcare because it was free and didn't always trust the doctor's advice. Each family was visited regularly by field research assistants who collected socioeconomic, surveillance, and food recall data. Sample collection and surveillance of cohort children began within 72 hours of birth. So meconium, the first stool of their lives, was entered into the study as their first sample that they submitted. Blood, urine, and fecal samples were collected regularly. So I think from the baby's birth to, I think through the first 3 years of life, sent to ICDDRB and then to the Gordon Lab for metagenomic analysis. During my time in Dhaka, 3 Bengali field research assistants introduced me to families in Mirpur enrolled in the Gordon Lab study. Children in their community suffered from extraordinary bouts of diarrheal illness, and malnutrition was often the result. The Gordon Lab was analyzing the microbial genomes of fecal samples from these babies to see what microbes they had and what the microbes were doing. I served as the lab ethnographer, gathering information on lifestyles, diets, and practices of everyday life. I want to give you a bit of an ethnographic snapshot here to think about how we can start to overlap the scientific and social concepts of health, how to use different kinds of expertise and data to do the most ethical and effective science possible. In this morning's session, folks were talking about populations and populations that are underrepresented in microbiome research. But the solution to this isn't just to sample from more diverse populations to have a wider representation. It's also to work with social science experts who are able to ask different kinds of questions, who can think carefully about who's going to benefit from the science. Where does the power lie? What are the social, political, economic conditions of life for people who we're studying? It's also critical to hear the voices of the people we're studying too. So this is Kanta and Rimi. These are all pseudonyms, so not their real names. Some of these families are still enrolled in studies, and a modicum of privacy is what I'm attempting. Kanta does not like eating. She's 18 months old and quick on bare feet out the open door of the nutrition center in the hot Dhaka street 3 times before her mother, Rimi, catches her. Typically gentle and affectionate with her daughter, Rimi holds Kanta firmly in the green light of the nutrition center, under illustrated posters teaching about nutritious food and hand washing. Kanta cries loudly as Rimi tries to spoon cereal into her mouth. Quote, she hates the pushti packets, but she doesn't like any food, so we force her to eat. Pushti packets are locally produced, ready-to-eat therapeutic foods, macronutrient meals containing rice powder, lentil powder, molasses, and soybean oil. The Bangla word pushti translates to nutrition in English. Although half of infants in Bangladesh weigh less than 5 pounds at birth, almost 2 pounds under WHO birth weight standards, it is when children get very sick that families seek help from ICDDRB, which has an influential historical presence in Mirpur. After local health workers diagnosed Kanta as malnourished, Rimi enrolled her in the microbiome discovery study. Quote, I thought my baby had a vitamin deficiency, and most importantly, I wanted medical care for her. It's very good for my baby because she was underweight and now she's in good health. I don't mind giving the feces and blood samples. They can use them to find if my baby has any diseases. They will know about it and they can treat it, end quote. ICDDRB is an international health research organization and also a free public hospital in Dhaka that serves hundreds of thousands of patients yearly. Credited with the discovery of oral rehydration therapy for the non-hospitalized, non-intravenous treatment of severe diarrhea, ICDDRB is known locally as the cholera hospital. During the monsoon, or what is called cholera season, the ICDDRB serves hundreds a day, patients that spill out into the temporary cholera triage unit in the parking lot, far exceeding their 350 bed capacity. To give some context, Bangladesh has the highest population density in the world, 2,700 people per square mile. This is a map of the neighborhoods that I was working in. Each one of those little rectangles is a home. In what ICDDRB considers the ultra poor areas, the population density is 10 times greater, or about 30,000 people per square mile. There's a very high burden of diarrheal disease in these communities. One field doctor told me that children who suffer from the most severe chronic illness can experience up to 300 diarrheal incidents before age 3. Kanta's fecal samples collected by Rimi are packed on ice, travel to the ICDDRB hospital, and are eventually flown back to the Gordon Lab. There, they are inserted into the guts of germ-free mice, which are then fed a specifically lab-made representative Bangladeshi diet, bringing together biological and social representations of life in Dhaka. Kanta lives in Block D of Mirpur 11, in a 3-room house with tin walls. In one room, Kanta sleeps with her mother and father, and in the other, her grandmother, grandfather, and their 9-year-old son. The third room is occupied by a renter and her young daughter. 8 people inhabit this 20 by 24 foot space, sharing the attached bathroom and narrow kitchen with 2 gas burners, a supply water pump, and pit latrine. Daily, government and NGO tankers truck water into the communities and fill underground tanks, which are connected to above ground pipes. Drinking water in Dhaka is notoriously and extraordinarily contaminated with sewage, agrochemicals, oil spillage, solid waste dumping, and untreated industrial effluent. While boiling can eliminate some contaminants, it does not rid it of high levels of arsenic and other heavy metals. In Kanta's house, there are no doors, no windows, no shoes. On her street, there are chickens and ducks underfoot and an open drain in front of every house. Rimi cooks food for the whole day before the gas service goes off at 9 a.m., preparing huge pots of rice, dal, spinach, and potato mash, and negotiating time at the stove with 5 other families who share it. The way in which Kanta's microbiome, her microbial self, comes into being is dependent on its social as well as its biological location. Along with birth, breastfeeding, and genes, excuse me, it's shaped by local practices surrounding water, chickens, and bare feet. Excuse me, I actually joked that this might happen during my talk, and now it's happening. The microbial populations in Kanta's gut are specific to the dilemmas that these bring into being. But how do we account for the social and environmental factors? We have yet to correlate or integrate ethnographic data about these social conditions into data analysis with metagenomic microbial genomes. Microbes that come from inside people's bodies are manipulated in the guts of mice as objects for understanding the structure of disease. Sorry. Why these specific Bangladeshi bodies are chosen for biomedical research and intervention brings into familiar oppositions, global and local, rich and poor, right? High income, low income, brown and white. Our biological selves come into being, function in the world, manifest bodily forms, and enact types of health and illness, all through the agency of microbes. If these microbes both affect and can be affected by who we love, where we live, and what we eat, if microbes make up our genetic and physical selves, aren't then our political, social, and economic selves also at least partly microbial? Malia, mother of 17-month-old daughter Priyanka, was receptive to the questions I asked, but sometimes would think them a bit too probing. Malia grew up in the house we sat in. She now lives there with her mother, husband, and 2 children. Her husband owned a small garment factory, and she embroidered so sorry, saris at night when the baby was asleep. She said, quote, "When I was a child, food was very tasty and fresh. Things like spinach don't taste good anymore, so my children eat less. These days, people use chemicals on plants like fertilizer, pesticides, and it makes food taste different. Soil has become very dirty and contaminated. In the past, there were small amounts of plastic, but now there are huge amounts of plastic contaminating the soil that the food is growing in," end quote. Like other mothers in the study that I talked to, Malia was very sensitive to changes since her childhood. She saw environmental contamination as an explanation for kids eating less and becoming malnourished. Packaged food is sold everywhere in the neighborhood and is expensive. She is acutely aware of her material and nutritional environments. She takes seriously the microbial education that the doctors have given her and her responsibility as a mother to protect her family from microbes. I'm so sorry. The FRAs, or field research assistants, teach the mothers the word jibanu, in Bangla, literally causative organism. Jibanu is not a word in everyday conversational Bengali, and not exactly the same as bacteria, microbe, or germ. For mothers, it became a concept bridging food, water, and places in the home to babies with diarrhea. This is an important point, as mothers were encouraged to think about themselves in new ways, about their bodies and their homes, their hands, their babies' mouths, and how their flesh touched pot handles, latrine buckets, and okra stalks. Mothers enrolled in the Microbiome Discovery Project began to think of themselves differently. They conceived of Jibinu as part of places in their lives, like markets, street food vendor stalls, their own kitchens and latrines, but also thought of Jibinu as part of themselves, as organisms they carried with them and passed on to their children. So gut microbes are studied and understood through their collective genomes and metabolic processes. Studies of human malnutrition vis-a-vis microbiomes are at risk for resulting in public health interventions that only focus on fixing the damaged or immature microbiome. Much of the Gordon Lab subsequent work has talked about probiotic and prebiotic interventions to fix an immature microbiome. But what are the ways in which we can account for the biology and social inequalities, like poor infrastructure, poverty? Can we see the social simultaneously and inextricable from the biological, not as metadata, but as crucial interacting data? Can we situate biomedical conditions in a socioeconomic context so we can design real-world action that cares for the whole self? This kind of collaborative work is critical, but also deeply difficult, frustrating, and sometimes painful. It contains imbalances of power, funding, and research goals. As I've talked about today, social determinants of health are refigured through a focus of microbes in DACA. Observations on the ways of living, caring for children, and feeding families, as well as larger analyses of structures of power, violence, race, and gender can help broaden the scientific view of study subjects. Sorry, I lost my place there. I paid careful attention to how the responsibility and compliance get assigned to specific bodies and thought about how to help reroute scientific and global health interventions down a more equitable path, one that accounts for the material, environmental, and social vulnerabilities that contribute to the making of microbiomes. After my work with the Gordon Lab, I have done subsequent work. There are a lot of really great interdisciplinary, no, I shouldn't say a lot. There are a few very good interdisciplinary working groups, such as the Microbes and Social Equity Group, that is bringing together experts in different disciplines to think about microbiomes in a biological and a social way. These are my field research. In 2021, some collaborators and I who were from all different fields, a microbiologist, a geographer, an evolutionary ecologist, and I wrote this paper about the future of an anti-racist microbiome science. This touches on a lot of what Dr. D'Amato was talking about in terms of how we think of race in these studies, and how we account for racism and how it impacts people, environments, and microbiomes. Excuse me. So this work was written for a scientific audience, but I feel like it has been eminently impactful and is one of the most meaningful things that I have worked on, because we're thinking about structural drivers of microbial populations and not just using race as a proxy to talk about different kinds of populations. And just the last thing I'll say is as a shameless plug is that a book that I wrote about all of my work with the Gordon Lab is coming out this spring from University of Minnesota Press. So just got into the catalog today. So that is my decade of work with the Gordon Lab. I want to thank the Gordon Lab and all of the subjects for allowing me to talk about their lives. Thank you.
Thank you, Dr. Benezner, and congratulations on your book. Well, those were fascinating talks. Thank you both, Dr. Benezner and Dr. Amato. We'll move to the 30-minute QA session now. So please continue to submit your questions and upvote questions that have already been asked. And I'll start with the ones who have been upvoted, and then we'll go in chronological order after that. So let's see. The first question looks like it may actually go back to Dr. Amato. It's a question from Ken Lassen. And the question is, there are over a dozen studies on microbiome shifts seen with long COVID. Are you aware of any microbiome intervention studies for this condition?
I am not aware of specific interventions that have been put forth thus far, although this is not my area of research, so there could be things out there I'm just not aware of. But I do know there's been a lot of interest, not only in long COVID, but also in terms of like severity of illness, in terms of thinking about, cytokine storms and this kind of runaway inflammation that you get and people that get really severe cases of COVID, and how that could be linked to essentially the body's propensity to kind of move towards inflammation, and the microbiome could be contributing to that, as well as contributing to some of the underlying conditions we know that increase risk of severe COVID, like obesity, diabetes, things like that I talked about in terms of being disparities. So I think there's thoughts about like, how can we kind of promote an anti-inflammatory microbiome more generally in terms of maybe diet or probiotics, things like that. But I don't know that we have a kind of silver bullet solution, which is true for all of microbiome research, essentially.
That makes sense. The next question was actually the most upvoted question for Dr. Benezra. The question was, do the participants get any insights back about their microbiomes? But I might couch that in a broader question of, how do you pitch a study? How do you recruit families into a study when there's such a different understanding of what you're looking for, and how do you explain that, and how do they benefit from this?
Yeah, that's a great question. So I can talk specifically about the work I was involved in the Gordon Lab, but in general, the concept surrounding microbiome is a very high income country concept. It's not a priority for people who are, battling with infectious disease or like constant enteric disease. In some ways, it's a luxury science, not to insult anyone here, honestly, but, it's a concept. So in the study that I was working in, all of the Bengali field research assistants, and I asked them specifically about this, did not talk about good microbes, microbiome that is important to your health. They only talked about pathogens. So the focus was only on hygiene, clean water, disease-causing organisms, because there was a feeling that it would confuse the issue too much if we started talking about commensals and good microbes and all this other stuff, which is really problematic, right? Because that's all that the Gordon Lab scientists cared about, was thinking about beneficial, I don't even know the right words to use, because as all of you as microbiome experts know, that there's no such thing as a good or bad microbe, right? It depends on the time and place and, all kinds of other surrounding factors. But there was absolutely no understanding that microbes could be important to your health. It was deliberately left out of the conversation. So there was never going to be any parity in terms of understanding what the science or the study was about. But on the other hand, something I often think about is all the children in that study lived, which is not true of everyone in that community. So that is a really important outcome, and was the mother's priority, obviously. Like, they really didn't care about the science. They cared about keeping their kids alive. So how to figure out how to introduce concepts that will be useful or valuable, what is the benefit sharing of microbiome science for these populations, is a very good question that not really very many people are tackling or taking on.
Thank you. Next question. Let's see. Again, for Dr. Benezra. This is from Christian Diner. What is your vision on unifying anthropological factors with the microbiome field that always tries to represent individuals by incomplete sets of quantifications, metadata that rarely describe the complexity of those factors?
Yeah, that's like my life's work to try to figure out the answer to that question. I think just having these conversations like we're having right now, having scientists and microbiome experts think about that this stuff might matter too, even though it's really hard to quantify and messy and is also very frustrating on my end, right? Like many times I've given talks to scientists who have asked me to quantify my data. They're like, well, put it in some kind of understandable set for us. And I can't, and I refuse to, I won't, because I did talk to 9 families. I can tell you 8 out of 9 families use toilet paper. I mean, is that a useful quantity for you? No, probably not, right? But the point of ethnography is that it's giving this very granular, everyday ethnographic picture of life that may be useful to you in how you think about your metagenomic data sets or something. Like there have to be ways where we have to learn how to talk to each other, because we really do speak different languages now. And have different priorities. Again, like Dr. Gordon and I both really felt strongly about developing a working interdisciplinary collaboration, and we still couldn't really figure it out. So not to be like a downer that it's not possible, because I feel like it's worth continuing to work on, but those kinds of data just don't even transact. But maybe we can make space for some more kind of ethnographic information in the scientific data, right? We can think very critically about how we're using categories of race. We can think about what are more precise ways to define populations, right? What are their toxic exposures? What are their diets? What are their all kinds of other social, biological, environmental conditions, instead of saying, this is what I always come back to thinking about like vaginal microbiome studies. The Revell study says Black women have these microbes, white women have these microbes, Asian women have these microbes. That's a very damaging way of thinking about microbiomes, right?
Thank you. The next question is in the same realm. It was asked of Dr. Amato, is by Christian Diner. I feel like many of the questionnaires used in microbiome studies do not capture determinants of health or inequities well. What do you think needs to change in order to correctly quantify the environment that we are sampling from?
Yeah, I mean, I think this is very similar to what Dr. Ben-Ezra was just saying. It's difficult, right? I agree that they don't quantify it effectively. I think first and foremost, and this goes back to some of what Dr. Ben-Ezra has written about, right? But why are we putting a certain variable into the study? Is that really the variable we want? Is it really race, or is it racism? Because it's probably racism, and in that sense, we should be measuring racism, and that can be experienced in a lot of different ways, right? And so I think trying to be very deliberate about how we're designing surveys and why. And I think this goes back to kind of, from my perspective, kind of a basic foundation of science is that we should have like a priori hypothesis about relationships between variables that could help guide some of that. And I think because the technology for being able to look at these microbiomes emerged so rapidly, and there was so much excitement in terms of just, trying to describe these communities, that at the beginning, a lot of that kind of fell by the wayside in terms of people just wanting to describe what they could find and kind of saying, this came from this place and that came from that place, and they're different, right? Because they eat different diets, but also like really, there's so many things that are different about those two places. And so maybe you only collected those diet data because that's what you could do, because there wasn't like this a priori hypothesis, because we had no idea what the relationships were going to be at the beginning, right? But I think now that we're starting to have an idea of like, these are some of the things kind of at a broad scale that are influencing the microbiome, to try to kind of go in and be a lot more deliberate in what we're measuring and why. And so, I mean, it's still difficult, right? We are never going to be able to capture that complexity entirely. But, if you're focusing on diet, making sure you're measuring diet carefully and being explicit about what part of diet you're interested in and why, right? I think that's the best we can do kind of on the scientific side, and also engage in these discussions that Dr. Ben-Ezra was talking about in terms of, here's what I'm thinking, but what's all the other stuff that's happening that I haven't even considered? And is there a way we could quantify that or not? And I'm a little different because I'm a scientist in an anthropology department, but I feel very strongly that like in my lab, if people are working on a data set, to the extent that we're able to make it happen, I want people to go visit that field site, because even though it's not the kind of detailed ethnography that Dr. Ben Ezra does, at least that's some sort of kind of qualitative input people can get that will help them interpret the data, right? Maybe it's not getting written into the results, but at least people can then say, here's the patterns we saw, here's the categories that it aligns with, but like, here's all the other stuff that could be happening in terms of like diet or access to water that we couldn't really quantify, but is probably playing a role, right? Or like, here's why that point might be out here. Like, I know that family was different and I couldn't really quantify why, but like, it makes sense that that family landed over here and this family landed over there. So I don't know if that was a great straight answer, because it is a hard thing, but that's what we try to do anyway.
And you touched a bit in your answer. Oh, Dr. Ben Ezra, did you have something to say? No.
I was just going to interject something, but I don't want to.
Go ahead.
I think that a really good example of what Dr. Amato is talking about is that, so again, the Gordon Lab is really interested in making probiotics, and we all want to solve the problem of childhood malnutrition, right? We can agree that that's the goal, to like try to fix that problem. They're really focused on a probiotic intervention, right? So I was always like this annoying voice being like, but people are still going to have open drains in front of their houses. They're still going to have pit latrines and terrible sanitation and no like government run infrastructural services. So you can give them lots of probiotics, but those structural problems are still going to exist. And so then their health problems are still going to exist, right? So just getting people to think about those as components of the problem, not as separate, right? Like not as peripheral, but as contributing to the problem is my goal.
Yeah, and who's going to make the probiotics and who's going to pay for it also are my questions.
And that actually segues well into the next question, which was asked by Dr. Gibbons about policy, which you did touch a little bit on a little later in your talk when you talked about, and this is for Dr. Amato, though you're both welcome to chime in. But specifically when you were talking about, maternity leave and how can we facilitate things from a policy level. But his specific question, if there's ways to elaborate, how might we craft policy to target and mitigate known microbiome adjacent inequities in marginalized communities? What would be the most impactful thing to do first?
That's a great question. I mean, my knee-jerk reaction is starting with some of the kind of early life parental leave interventions, because I think that's where a lot of these disparities in microbial exposure is set up and then is perpetuated. So I think, making sure that people have the infrastructure they need to be able to have a healthy pregnancy and increased chances for vaginal delivery, breastfeeding, that they can take time to like think about the nutritional input their kids are getting and kind of what it means versus, just making sure that they're fed, water quality, all of those things. I think targeting kind of that early life period would be a good starting point. In some ways, I also go to that because there's a lot of kind of policy that's trying to target that area to begin with, right? And then the trick becomes kind of what does microbiome research add to that, right? Like we already know that parental leave policies in the US are like terrible. And so why do we need microbiome data for that? And, we know that breastfeeding is good. And when people can do it, they should do it. Why do we need microbiome data for that? But I do think there are aspects of microbiome research that can kind of add on to that, right? In terms of like, with breastfeeding, I know there's people thinking about like, how different is it to like actually breastfeed versus give an infant pumped milk from a bottle, right? They're still getting the nutritional input of breast milk, but what happens in the microbial community when it's kept in a refrigerator and then reheated? We know that there are like daily fluctuations in what breast milk looks like. So does it, how much does that influence the infant nutritionally and microbially if you're giving the infant, milk that was pumped in the morning at night? what is the infant getting by being like actually in contact with mom physically versus being fed from a bottle by somebody else? Those are things that I think can enrich perhaps attempts at improving policy that is currently, either in existence or being developed. So that's where I would tend to go. Although there are so many things that could be fixed about environmental disparity, it's kind of hard to pick one. I think food deserts would be another, access to high fiber, like fresh produce would be another place I would go immediately, since diet has such a strong impact on the microbiome.
And you even mentioned in your talk the circadian rhythms and shift work and policies that could cause protections in that as well. So.
Yeah, no, so many things, parks. How are we building parks? There's all these new fancy parks being made out of like the rubber stuff and metal, right? But like maybe sand and gravel is better because it's an environmental exposure, right? So, yes, we're building parks, but how are we building them? Out of what materials, and how does that affect microbial exposure? So yeah, there's a lot of different directions, but I guess where I would go first would be early life, since that's when the microbiome is getting established. And you're gonna like set up disparities for sure at that point. I don't know.
Thank you. The next question is for Dr. Benezra from Dr. Gibbons. Thanks for that beautiful talk. I think it's critical to understand how we can create fruitful collaborations between social scientists, anthropologists, and biologists. What were the biggest impediments for making this work well in your experience?
So many. But I don't want to sound like discouraged or not hopeful. I think that the kinds of solutions that we each want to come up with. So when I teach my students about this kind of thing, we talk often about Paul Farmer's ideas of proximal and distal interventions, and that they don't have to be mutually exclusive, right? You can think about structural things and immediate interventions as simultaneous and so often, like, especially global health has a really specific intention and way of doing things. And social scientists, I'd be the first to admit, we can be super obnoxious and really critical, like we're the ethics police and we just are like really good at pointing out what science is doing wrong. And that is not helpful either, right? So figuring out how to work together and be like a willingness and generosity is extreme. Collaborations are all about individuals, right? They're about like individual people trying to work together. So I think that so for me personally, the biggest impediments were that when I started working at the Gordon Lab, I was a PhD student, right? I was a PhD candidate, and Dr. Gordon, as you all know, is the father of microbiome. So there was a huge power disparity that was never going to be resolved, understandably, right? And so the ways in which, science wants to solve problems and anthropologists want to solve problems, and what those problems are really difficult to resolve with one another. And so that is really, and how to talk about the science and what the priorities are really, really difficult. And again, let's be honest, science funding is always going to be more than social science funding, right? There's always going to be a power and money dynamic there. And I think that we have to just keep trying to talk through what is actually important. But getting scientists to value qualitative data is probably the hardest thing that I have to do. To be like, this is not just something nice for you to know about, that this is affecting microbiomes in a really important way, and will make your science better and will make it more effective and more precise. And so that can be frustrating for social scientists, and then we just give up. So social scientists need to keep going as well. But I've been doing this work for a long time, and it's very hard for me to get funded because there are a lot of disciplines are institutionalized in terms of journals, in terms of grants, in terms of jobs, right? You can't be an interdisciplinary like free-for-all. There's no faculty positions that are like interdisciplinary free-for-all, right? You have to be in a discipline to get a tenure track job. So, or to publish in a journal. So, you know, places like M Systems are thinking more about who's publishing in their journal. Like all of this equity stuff also has to do with who's doing the science, not only who are the scientific subjects, but who's doing the science, who are the editors, who are the reviewers. All of that is also super important. So as those boundaries start to break down a little bit, it becomes easier, but anthropology organizations never want to fund me. They're like, that's science, you're way too sciencey. But also scientific organizations don't want to fund me because they're like, what are you talking about? That's nonsense. So, you know, where does that leave me? Hopefully invited to symposiums like this, which is great. But I don't know if that answers Sean's question at all, but.
It does. Thank you. Next question is for Dr. Amato. Can you please discuss a little bit about the complexity of diet assessments that include underrepresented populations and how this may affect microbiome research? So I feel like both of you have had experience with this, if you'd like to add.
Yeah, I mean, I'll start by saying, so Katie, as you said at the beginning, I started in field primatology, right? That's my initial background, and looking at feeding ecology and diet and the microbiome in that context. And a lot of the kind of general principles and relationships I'm interested in I think are transferable to humans, although humans are much more complex in terms of lifestyle variation. And so I struggle with this question all the time because it's so much easier to get data from wild primates. People are like, how do you do that? I'm like, well, I do have to go out in the forest and follow them around, but once I'm there, I literally follow them around all day and write down every single thing they ate, and I pick it up and I can analyze what's in it. And that's harder to do with people. It's possible to do, but you can't do it with very many people, and it's super invasive, right? People don't want you following them around all day necessarily, like writing everything down they do. And so, you know, we rely on surveys often, and I think there's so many potential problems with these surveys, right? In terms of, you know, like I said, what part of diet are we interested in? So a lot of times these dietary differences are being reduced down to like macronutrient differences or fiber differences. And it's something that I've done also, because at a certain point you have to just decide how you're going to categorize diets. And even if you have a super detailed dietary survey, you can't put like 100 different questions about diet into a quantitative spreadsheet and get something out of it, right? Maybe one person eats this, one person eats that, and I can't say anything about that, even if I think it might matter. So I really struggle with how to best do the dietary data. I mean, there's also questions, sorry, I could talk for hours about diet. You know, are we worried about like 24-hour dietary recalls, like what the person ate in the past 24 hours, or kind of what their general pattern is overall? Like, usually I eat this, usually I eat that. It seems to me like often the latter is more related with patterns that we see in microbiome data if we're going to have to categorize and quantify. But that also leaves a lot to be desired in terms of kind of detail here. And in terms of like the spices that Dr. Ben-Ezra was talking about and kind of how food is prepared, like that gets left out a lot, right? It's like protein, lipids, carbohydrates, fiber. But I think it matters if people are making that in their house, if they're making it with oil, if they're going out, right? And we try to capture these things in surveys, but it's always going to be imperfect. So again, I think I would go back to, I think it's currently oversimplified. I think it will always be oversimplified because it has to be at some level to quantify it from the kind of sciencey perspective. But I think making sure that you have clear hypotheses about what are the relationships you're interested in, right? Are you interested in fiber for a particular reason? And what are the other things that could be interacting with that? So that's kind of my best attempt at dealing with that. But I know there's been people also exploring, I'll just say briefly, beyond looking at macronutrients, looking at like metabolite profiles of the food, or kind of trying to get like more detailed. And in some respects, that's getting past the foods, and in some ways could be more standardized, but I still think you run into the issue of like how it was prepared. I'm not sure the extent to which that would come out in like metabolite profiles. So yeah, I'm not sure I don't think anybody has a good answer, but that's how we try to deal with it.
Dr. Benezra, would you like to elaborate on how to actually study humans in fieldwork? You actually have done that.
Yes, that's true, but I didn't write down everything they ate every second of the day. And I know that when I was in St. Louis, they tried to do a twin study where they had people stay in like a hotel room for 5 days so that they could control their diet. And I think half of the people dropped. They were like, fuck this. Like they were like getting paid thousands of dollars and they were like, no, thank you. I can't do it. Right. Human studies are so difficult in that way. But I think that what Dr. Amato is saying, in just even like accounting for, it's not just maybe components of, nutritional components or like the biochemistry of the food, but also all the other things that go, in DACA, families share cooking pots and also share kitchens with 5 other families, it's going to have an effect on the food that they are cooking and obviously the microbes as well. And so, yeah, the diet surveillance in the Bangladesh study was done by the field research assistants, and it was like a food recall. Like, what did you have for the last 24 hours? Which obviously people are terrible at remembering things, as we all are. Like, I could not tell you what I ate in the last 24 hours. So maybe thinking about how important is that precise, detailed information, right? Like, what other kinds of things might it be more useful to know? I don't know.
Thank you. I think we have time for one last question. There's a question that applies to both of you. It was asked of Dr. Benezra, do you have any advice for quantitative scientists wanting to collaborate with social scientists or vice versa?
I mean, it's awesome that people want to do that. And I guess thinking about because I think part of the problem is that when you're trained as a microbiome scientist, you're not trained in all these things, right? No one expects you to have expertise, just to seek out expertise of people who might have a doctorate in studying humans and culture and things like that, right? So I guess being aware of what you don't know and seeking out an expert who might know it, right? And just accounting for these things as important, right? Because there are anthropologists who are experts in every single country on Earth, right? And every region of every country on Earth, and could tell you a lot about the very specific diets of people in very specific places. But yeah, we never connect with those people, or microbiome scientists aren't thinking about that. So I think just even asking that question about who else could I add to my team, right? Or my research design, or thinking when I'm writing a grant, I mean, anthropologists are super cheap. We really do not, we don't have labs. We don't have a lot of specialized equipment. We have notebooks and tape recorders, and we're very inexpensive. So, next time you're writing a grant, think about, how can I very cheaply add a social scientist that will enrich my data in these ways, right?
Thank you.
Yeah, I would second that. And I think the other thing that I would add in terms of I haven't had a collaboration over a long-term project, but I've done little kind of papers or panels here and there. I think the point that Dr. Benezra made earlier about open-mindedness and also understanding that the language is really different, right? So reaching out to people about collaborating, but then realizing that it might take a little bit for you to actually understand each other. And then the other thing that was brought to my attention, and I've kind of been thinking about, is also what do each of you need out of the project, right? Because I know there's also sometimes, I think, although Dr. Benezra, you can correct me if I'm wrong, there's sometimes a hesitancy maybe from some of the social cultural anthropologists in terms of, you don't want to just be the ethics police or the here's how you define this, right? You have your own research goals and your own, kind of career milestones, right? That you have to, that you have your own vested interest in things, and that might not always be the same as it is for a quantitative research scientist on the other side. And so making sure you can both get something valuable out of it, I think, is also an important discussion to have, so that it doesn't feel like one person is kind of taking advantage of the other necessarily. I don't know.
Great. Well, thank you. We're out of time now, so I'll go ahead and conclude our Q&A session with another warm thank you to Dr. Amato and Benezra for giving us such a grand finale to our symposium today, as well as extend another round of virtual thanks to all of our fantastic speakers. And with that, I'll return the floor to Dr. Gibbons for closing remarks.
Thank you.
Thank you.
Thanks.
Wow, what an incredible day of talks. And what great questions from the audience. Thanks so much for your engagement throughout the day. Thanks again to our sponsors, Illumina, Metabolon, and the Washington Research Foundation for allowing us to make this open and free to all. And I huge and special thanks to all of our speakers and session chairs today. Our chairs, Christian Diener, Noah Rappaport, and Catherine Stevenson, masterful jobs today, fielding questions and interacting with the speakers. This was really great. The first session, it was really exciting to see people, extending microbiome research across the globe. Dr. Poirier and Dr. Irola showing us great examples of that. In the second session, we got sort of a more focused view into a particular problem, this curious case of the genus Prevotella, with Dr. Luzapon, and we were hoping to get Dr. Jimenez, our second speaker. Sadly, we lost her. I hope she's okay. Maybe I think the internet cut out or something, and so I had to step in there to give a talk. But hopefully we can get Dr. Jimenez back some other time. And the third session was just wonderful. It was focusing on the social determinants of a healthy gut and thinking more from the side of social sciences and anthropology, and as quantitative scientists, how we need to think qualitatively about the systems we're studying, think more holistically and carefully about what we're quantifying and how we're quantifying it and how that relates to the underlying data. So just super pleased with today's symposium. So specifically, I need to also thank again our instructors from the course, days 1 and 2, Christian Diener and Nick Bowman, really amazing tutorials. Our guest speakers from the course also gave great talks the last couple of days, Dr. Ashish Jha and Alex Carr. Our teaching assistants during the course provided you with help on Slack. Those who were there were Noah Rappaport, Alex Carr, James Johnson, and Samantha Picos. Our administrative support, without these folks, none of this could have happened. Thanks to Dominic Lewis, Allison Kudla, Joe Meister, Audrey Hubbard, Connor Kelly, Victoria Uhl, Shanna Braga, and Thea Swanson, and really the whole ISB facilities team for pitching in and helping out here. Finally, we'd really like to thank our DEI consultants this year. We were a little afraid of having this as a theme of our series this year. Most of us are from the developed world, and white, for example. I'm a white man, and so we wanted to make sure we were thinking carefully about how we were presenting this material, and we were able to work closely with the Microbes for Social Equity group, and in particular Emily Vissel and Dr. Sue Ishak, who helped us kind of craft the content for our program this year. Again, as a nonprofit research organization, we rely on the generous support of donors and sponsors this year, for example. If you happen to have a little extra funds in your pocket and you want to support the kind of work that we did this year for next year, there is a donate button at the bottom of our website. If you want to learn more about us and about the Institute for Systems Biology, you can visit us at isbscience.org, as you can see over here. If you want to share your experiences from the course and symposium, you can hash us on social media, hash ISB Micro 22, and you can tag ISB so we can give you a reshare at ISB SCI. And with that, that's a wrap on the 2022 ISB Virtual Microbiome Series. I hope those of you who joined us these last 3 days have enjoyed it. I know we've really enjoyed being here with you these last couple of days. Thank you so much for your time, and have a great rest of your day.